Okay, good morning or good afternoon or good evening, depending where you are, but welcome everyone across the globe. Thank you for joining us and a very warm welcome to this session on solutions to pollution, contaminant impacts and remediation in Antarctica. Firstly, I'd just like to introduce myself. My name's Catherine King. I'm a Principal Research Scientist at the Australian Antarctic Division, and I'll be chairing this session. I'd also like to introduce my colleagues, Doctors Catherine Brown and Jane Wasley, who I work with at the Division as co-conveners. There was a last-minute change in the program. Two other colleagues that were supposed to join us for this unfortunately couldn't make it. So a special thanks to Jane and to Catherine for stepping at the last minute to help out. And together with the IT team on the ground in India, we hope to deliver a relatively smooth session to you today without too many IT hiccups. So just a bit of an introduction to this session. We're all working in this field, obviously, and this is probably background that you already know, but I'd just like to put it into context for what we're going to hear about from various speakers. So Antarctic terrestrial marine ecosystems, as we know, are subject to human disturbance at global, at regional, but also at local scales. And as we continue to increase human activities in Antarctica, and these continue to grow, the stress on Antarctic ecosystems and the potential for environmental impacts obviously increases. So this interdisciplinary session will include research on various aspects of pollution in Antarctica, on input pathways, on environmental drivers of contaminant dynamics, on some of the sources and the fate of contaminants, on the effects of contaminants on biota, and importantly, on mitigation and remediation solutions. So here we have a platform to really quantify the scale of this problem in Antarctica and to propose solutions for minimising the environmental risk and the impacts of contaminants on sensitive Antarctic ecosystems. So for this session, this is actually the first of 3 sessions on this topic. We have 2 sessions today, a 2-hour block. We'll have a quick break and then the second 2-hour block. And we also have one tomorrow at the same time as this, followed by a 1-hour discussion, which I really encourage you to come along to. During this discussion, at the end of that third session, I'll also invite any of our e-poster authors who'd like to present their poster to the group. So that gives them a bit of a platform. Unfortunately, we couldn't schedule that in, but I think that's a great opportunity for those authors and researchers to present their work. If you haven't been to the e-poster exhibition space on the events platform, it is up and running, and you can browse those e-posters at any time throughout the duration of the conference. And you can also comment on them and or ask some questions which the authors will address. And I think we have 12 posters for this session for you to check out. So I encourage you to do that. Just some housekeeping before we get into the nuts and bolts of the session. Each speaker will be allocated 10 minutes, and that'll be followed by around 2 minutes of questions. So please, speakers, keep track of your time. We'll also let you know if you're running over time so you can wrap up. On that note, I should comment I might be running over my 3 minutes here. We have had a speaker withdraw in this session, so don't panic. We will still run to the schedule as it is, as shown on the screen as well. Please, with your questions, everyone's on mute apart from the presenters. So we're asking you, if you haven't been to any SCAR sessions so far, we're asking you to type your questions into that chat box function within Zoom, and then we will keep an eye on that, and we can address those at the end of the presentations. We will have that short 2-minute presentation, so short 2-minute block for questions at the end of presentations. But if there isn't time, we also have 20 minutes at the end of this session where we can have a more open discussion. Everyone except the current presenter and conveners should be muted at all times throughout the session, except for at the discussion at the end of the session, as I said. Please note, you will have already heard this, I hope, presenters, but this will be recorded and available via the SCAR virtual platform in the past events sections once this session is finished. It'll also be available via SCAR's YouTube channel, and I think that's within about 24 hours of the session end. If for any reason you don't wish to publish your talk on the platform, please let us know at the start of your talk, and the IT team will pause recording. And similarly, if you don't want your presentation to be made available on the SCAR YouTube channel, please send an email to opt out to the SCAR organising committee. I think that's by this Friday. All right, I think that's enough housekeeping. Let's get into it. We're pretty well on time. So in this first of 3 sessions on this topic, we have 7 speakers. Unfortunately, we had a last-minute withdrawal from our second speaker, which is Melanie Borup, due to illness. So she'll be out of the program, but we'll keep Andrew Klein's talk set for 3:27. So we'll keep to that timetable. We'll just have a bit more time for questions or time for a tea break after I speak. So, all right, let's get going then. So I'm starting the session actually, so I am the first speaker. So I'll just keep going here and roll off to get us started. So as I said, my name's Catherine King from the Australian Antarctic Division, or the Australian Program, and today I'll be describing some of the work done by the Australian Antarctic Program on the management and on-site remediation of fuel spill sites at subantarctic Macquarie Island. I'll particularly focus on the impact.
Oh, we don't have your slide up yet.
Okay, it's already gone wrong. Let's try that again. Sorry about that. Lucky we have a gap in the program. Thank you for stopping me. I appreciate that. How are we now?
Good to go.
Awesome. Thank you. Thanks so much. Okay, so this presentation describes some of the work the Australian program's been doing on the management and on-site remediation of fuel spills at subantarctic Macquarie Island. And I'll be focusing here on the impacts of hydrocarbon contaminants on native biota and the development of screening level environmental guidelines and remediation targets. As most of us here are likely very familiar with, there are numerous examples of localised contamination that affect our station areas. Many of these are a legacy of past environmental practices and poor waste management, but we also have contemporary issues that we're dealing with. These are particularly to do with wastewater discharges from stations, as well as accidental fuel spills. As we still largely rely on diesel for power generation, and as our station infrastructure is ageing, there's an ongoing risk of spills associated with the use of fuels, with fuel transport, and with fuel storage. So our approach to the management of contamination at the Australian stations is multidisciplinary. It's really about integrating chemistry, biology, environmental toxicology, and engineering for evidence-based protection of the Antarctic environment. Through our site assessments, we seek to understand the chemistry and the ecology of the system. So what is the contamination? Where does it occur? And how much is there? Our risk assessment process includes biodiversity studies and environmental toxicology. So what are the biological communities or the receptors in the receiving environment? And how sensitive are they, and how are they actually affected by contamination? These then inform prioritisation of sites and engineering solutions that we use to remediate these impacted sites. So Macquarie Island, for those of you who aren't familiar with it, is located in the Southern Ocean, approximately halfway between Tasmania and eastern Antarctica. And the station is located on that small isthmus at the very northern end. Here's a view of the current station, highlighting 2 of the main areas of contamination that we've actively remediated: the fuel farm, where fuel is stored, and the powerhouse, where fuel is used for power generation for the station. And most of the contamination we're dealing with here is from special Antarctic blend fuel, which is a diesel derivative that's formulated for use in cold climates. This slide shows a conceptual site model and cross-section at the fuel farm as an example of a site we have assessed and remediated. And it illustrates the complexity of the site, showing those multiple sources of contamination, some of the key receptors, both human and ecological, as well as contaminant migration on site and the connectivity that we see from the land to the marine environment via that groundwater flow. Our remediation program on site has involved a range of technologies, but the main ones really being in situ aeration and nutrient enhancement in the soil, and those are shown here in this photo. So these processes or these technologies increase the rate of microbial activity and thereby the natural biodegradation of hydrocarbons in the soils. So this is important, obviously, as in polar regions, these processes are so very slow. And we also use permeable reactive barriers for in situ containment. This in situ remediation has proved really successful at speeding up the reduction of total hydrocarbons in the soil, but we also need to understand how biological communities would respond and how they recover, and to answer that vital question in any remediation program of how clean is clean enough. So to help answer that question, we use toxicity tests. So we add known amounts of a contaminant to biota, and we estimate what concentration of that contaminant will cause harm. In toxicity tests, we'll therefore expose a population of organisms to a range of concentrations of the contaminant and look at their response. And this response may be in terms of reproductive output, germination for plants, or survival, for example. And from this, we then fit a concentration response curve and derive what is known as critical effect concentrations, or CECs, which are predicted to affect a certain population, a certain proportion of that population. And some of the main CECs that we use are listed on the right-hand side here. This example plot shows the modelling of an EC20, which is a concentration that is predicted to cause 20% decrease in the response of the population relative to the control. As all species have different sensitivities to contaminants, we obviously need critical effect concentrations from a wide range of species to best represent the response for that given community. So the environmental guideline derivation process combines all that data from multiple species into what's known as a species sensitivity distribution model, or SSD model, as shown on the right. And from this SSD, protective concentrations are generated. And these are the concentrations predicted to protect a certain percentage of species within a community. So for example, a PC95 is predicted to protect 95% of species in that community. And in the risk assessment process, the selection of an appropriate PC value at a site is based on the level of protection thought to be most suitable or appropriate for the land use at that site. So for our contaminated sites within the station footprint at Macquarie Island, we'd use a PC80, for example. For a more pristine environment or an undisturbed site, a PC95 or even a PC99 might be deemed more appropriate. So that's a bit of theory behind it, but what's the sort of data that we have to work with from Macquarie Island? We've done a number of studies over the years on the effects of fuel contaminants in soil to native terrestrial biota, and this has included work on invertebrates, on plants, and on microbial communities. And these tests have ranged from traditional single species tests, such as with the earthworm or the native grass, through to functional and community-based tests with microbes. And we've also tested soils with different properties, so including a range of different organic carbon contents and different ages of fuel contamination and therefore hydrocarbon degradation. And these properties represent the range of soil types that are occurring at that site. So we pulled all that data from those tests together and compiled them to form a comprehensive list with around 214 CEC values. The studies obviously used different methods and derived a range of different critical effect concentrations, which we then converted and standardised to EC20 values as required. And then we employed an expert judgment approach to select the most robust endpoints to use to construct our SSD models. And the criteria that we used included the quality of data and the degree of confidence around those CEC estimates, the biological relevance of the endpoint to the species in terms of population growth, the ecological relevance to the overall soil health and ecology, and the relative sensitivity of different endpoints within a species. And from this, from that expert judgment process, we then selected a subset of 13 values which were used for inclusion in the SSD model. And this was then used to produce that site-specific soil quality screening guideline or remediation target for contaminated sites within the station area. So here are our 13 data points plotted in SSD, and you can see the different invertebrates, plants and microbes each have different sensitivities to fuels. And this species sensitivity distribution curve is fitted to these individual data points with those 95% confidence intervals. And from this model curve, we can then determine protective concentrations at different levels. So shown here in red, we have the PC80, and this is of relevance to our remediated sites in disturbed environments within the station limits. So this then becomes our sort of magic number that we have that can answer the question, how clean is clean enough to protect biological soil communities at the site? And this number becomes our remediation target. So on the site, we then apply this P80 value to our site assessment work. So this shows an example of how one area at Macquarie Island, where we have used the guideline value, the PC80 of 105 as a screening criteria. So the red dots show you the sampling points in which fuel concentration was measured in the soil. And the green areas are indicative of where those concentrations in soil exceeded that PC80 guideline value. The yellow indicates soils with potential human health risks as well. So by comparing those concentrations measured in the soil on site with that PC80 value, we can really see where locations where there might be a need for further consideration of risk to the environment and where further remedial action might be required. So just a few conclusions from this work. The toxicity studies and sensitivity estimates for local biota are absolutely necessary for the derivation of site-specific environmental guidelines in Antarctica. And these guidelines are then critical as a screening tool to evaluate risk to biological communities. The approach that I've described here and that we've used is recommended and will be used in our assessments of other contaminated sites at Australian stations in Antarctica. The robust PC80 value of 105 milligrams per kilogram of fuel that we derived is likely to be appropriate for similar environments, including other sub-Antarctic islands. And Antarctic sensitivity data is very limited and needed. So what I presented is, after 20 years of research in environmental toxicology by the Australian program, this really represents our most comprehensive data set and is actually the first time that we've really been able to derive a robust value and apply a target value in a site assessment. I just want to give a plug to our new research program, A Cleaner Antarctica. So Australia is just about to embark on a decade of research to assess all our contaminated sites across our stations to identify priorities for cleanup and remediation. So we will further investigate the sensitivity. As part of this, we'll further investigate the sensitivity of native biota to contaminants through toxicity tests, and we'll continue to develop appropriate remediation technologies. We'd especially like to hear and engage with people internationally and other nations that are also addressing these issues. So we'd be very keen to hear from researchers that are working in this area. That's about it from me then. So thank you so much for your attention, and I'll throw it open to questions, please. So if you have questions, as I mentioned at the start, if you can add those to the chat screen, that would be great. We have quite a bit of time now because Melanie, our next speaker, has unfortunately can't join us today. So we have about 7 minutes So I do encourage any questions and any discussion points that people have. I'd love to know if anyone in the audience has used a similar approach, for example. Okay, so there's a question here from, oh, sorry, Kath, do you want to field those?
Oh, I see, yes. So from Andrew Klein, we have, so how large of an area had TPH values in excess of your EC80 value?
Okay, thanks, Andrew. Yeah, so it's a PC80, protective concentration. So that's the green shape that I drew around that was centered around one data point in which we had an exceedance of that PC80 value. So we're being quite conservative in our estimates of the area of coverage. That was one site amongst many. We have maybe 5 or 6 of those similar diagrams where we've had, or similar maps where we've done the chemical measurements in the soil and then cross-reference that with our PC80. So to answer your question more specifically, we're talking about several metres. We're not talking about huge areas. And as I said, we're being conservative and it's based on, generally it's based on 1 or 2 exceedance samples. Does that answer your question? So generally we are hitting below the PC80 in many sites. Yeah. Great, thank you. And where we're not, I should say, there does become a limit to how much remediation you do at a site. And part of our investigations in looking at the response of biota with the plants in particular and the native grass, we found that it was the best phytoremediation tool that we could hope for. So it really wasn't good for phytotoxicity. It hardly responded, but what it did do, it did reduce the concentration of hydrocarbons in the soil, and it was really a remediation tool. So some of these areas where we are now, where we do have these exceedance values, probably one of the approaches that we're looking at, and probably the best approach, is actually to revegetate those areas. So put in those power plants, the native grass, which actually are natural biodegradators and really clean up that soil for us.
We have another question and comment from Linda Goldsworthy, who says, excellent presentation. Are you aware of any other parties doing this sort of work? And how do you ensure no spread of the use of live test toxic contaminants?
I can't see those questions, Kath, sorry. So the first question was, I don't know why.
They're in the Q&A.
Okay. Yes. Okay, great. Okay, if we can try and keep to the chat, but that's fine. Thanks for the question, Linda. I've got that now. Not many people do this toxicity test approach. So certainly in Antarctica, certainly it's used heavily for regulation and for discharge of wastewaters or for contaminated site management back in Australia and Canada and the US, around the world. Not so common in Antarctica because it's not required, I guess. There is some, and I'm hoping there are people within these sessions over the next couple of days that can say, yeah, we are working that area, because it has been hard for us to find other toxicology work other than looking at body burdens and things like that. Not many people do this experimental approach, and I'm not aware of people ever generating these remedial targets or remediation targets or guideline values that they then use across the site for site assessments in Antarctica. So to my knowledge, no, but I would love to hear it. Our team here would love to hear if people are. So thank you for that question and highlighting it. How do you ensure no spread of the use of live test toxic contaminants? Okay, I should have said that we don't do that. These are not in situ toxicity assays. They are done under laboratory conditions where we are trying to mimic what would happen. So the test conditions are trying to mimic what happens in the natural environment. We don't actually go and release TPH into the environment to do these tests. They're laboratory based. I think that's what you mean by live test toxic contaminants. Is that correct?
We have a couple of other questions and comments from Dr. Rahul Mahan. Can this be circulated as best practices?
Thanks for that question. Yes, so you guys are probably aware of the cleanup manual, the CEP cleanup manual. So not just us, lots of researchers that are doing this sort of work in Antarctica contribute to that. This is, as I say, it's the first time we've had it. We've amalgamated enough data to actually construct that SSD model and to test this approach. It is absolutely our intention for this to be incorporated as best practice and be submitted as a methodology or as a recommended approach for contaminated site assessment, and we'll be submitting that to the CEP in future years. So we're still going through the final stages of this. It's not published yet, but absolutely, I think this should be circulated as an appropriate best practice. I think we need to wrap up there, but great discussion, and hopefully at the end we can talk more about these. But I think we're about time to hand over to our next speaker. Thank you for filling in that gap there with Melanie not here. So I'd like to introduce Dr. Andrew Klein, who will be talking about McMurdo and Palmer stations in Antarctica and environmental footprints. So please go ahead, Andrew.
Give me a second to get my screen shared. I wasn't expecting it that fast of a transition.
Sorry about that. Yeah, I'll try and keep us as no drama. We have time at the end, Andrew, but yeah, I'll try and keep us on track as much as possible.
I hope people can see my screen. Can people see my screen?
Yes, sir. Full screen, please.
Oh, I thought it was in full screen. Sorry about that. Okay, come on. There we are. That should be full screen.
Thank you.
For many years, my colleagues and myself at Texas A&M have been conducting environmental monitoring research at McMurdo and Palmer stations. And the purpose of this talk is to try to put this research in the context of a lot of recent work on defining environmental footprints around Antarctic research stations. So really beginning in 1999 and continuing in 2015, we conducted environmental monitoring, primarily at McMurdo Station on Ross Island, and also some at Palmer Station on Anvers Island on the Antarctic Peninsula. And if you look at the range of the size of scientific stations across the Antarctic, you can see that McMurdo is kind of a beast of its own, but Palmer Station is relatively among the typical sizes of Antarctic stations. So in recent years, there have been a number of publications by Sean Brooks and colleagues that both defined a set of environmental footprints, as well as mapped comparable measurements of some of these footprints across all Antarctic stations. So what I'm presenting here is some preliminary work that tries to place our extensive monitoring work into this theoretical context, as well as to try to develop a methodology that looks at trying to see if we can kind of create comparable footprint metrics for a variety of disturbances that we've looked at over the years. And I'm going to look at today some physical disturbance of the surface, contamination, as well as infrastructure. And then what I'm not going to have time to talk about, but also looking at, is how you take work that's been done at a fairly well-studied base, McMurdo, and transfer that to a less well-studied station, in our case, Palmer. So in a 2018 publication by Brooks and others, they described or proposed a set of definitions for environmental footprints. And I've actually personally found this really interesting because for years, our project has been trying to do this for McMurdo based on some pretty extensive measurements. So for example, we've tried to look at the physical disturbance of the surface as a way to look at the historical development of McMurdo Station over time. And that work's been published in a couple different places. And we've also spent a lot of time looking at chemical contamination of the soil based on several thousand soil samples that we've collected over the years. I'm going to talk about a subset of those. And what we've tried to do is to use various GIS techniques to go from specific soil samples into creating a map or maps of environmental contaminants. And this is total petroleum hydrocarbons for McMurdo Station, and parts per million is milligrams per kilogram to be consistent with the other talk. And what you can see about a spatial footprint is there's both an extent, the colors in the map, as well as a magnitude or intensity, in this case, the hue. And you can see some of these are well above the 100 and some that was mentioned in the previous talk, and I'll come back to that in a second. So these are useful, but how do you compare the environmental footprint for different chemical contaminants, as well as compare it to the footprint of infrastructure, et cetera? The work I'm going to talk about is based on very old GIS techniques that actually predate computers. And it goes back to work on what we call cartographic overlay today, which was developed by a very famous ecological planner called Ian McHarg, who in his famous book, Design with Nature, not only laid out the idea of ecological design, but also laid out the fundamentals of GIS. So what I'm doing is not necessarily all that new and innovative in some ways, but I'm applying it here. So the idea is we lay a grid over the surface, and this is McMurdo Station, and Scott Base is a little to the east of this. And this is actually a sampling grid that we based about 10 years of sampling on. There's about 4,000 of these 25-meter diameter cells, and we would randomly select some of these for soil sampling. But this is a grid I'm going to use in trying to figure out which of these cells is disturbed or has an impact and which doesn't, and also look at, in total, how many separate impacts are in these cells. I just want to note that one of the things we have to think about when we talk about any environmental monitoring or defining the footprint is there are places we don't measure. So we specifically didn't collect samples because it was either snow surface or it was too steep for us to get to, for example. So there's places we actually don't have information, and we need to keep that in mind. If we look at the physical disturbance I showed in a map a few slides ago, what you find is that about 93% of the area in this grid actually has some physical disturbance of the surface. So as a first cut, this area is highly disturbed. The methods I'm going to talk about is I'm going to try to translate a number of selected impacts onto this 25-meter hexagonal grid. And I'm looking at infrastructure and chemical contamination for this particular talk. And for selected infrastructure elements, buildings, fuel tanks, pipelines, road disturbances, a grid cell was considered impacted if it intersected one or more of these particular features. And I'll show you a map in a second. We also looked at chemical contamination of 17 different chemicals. And we're considering, for the purposes of this talk, a cell to be impacted if we found the contamination to be, or the levels to be 2 standard deviations above the mean concentrations that we measured at a number of off-station control sites. And we selected a threshold of 30 milligrams per kilogram for TPH. And it's interesting, so some contaminants are very patchy and they're very hard to interpolate. So for the 2 most common elements of contaminated elements we found at McMurdo, which were TPH and lead, you can actually take those samples, use GIS to create an interpolated surface that you can then intersect with the hexagons to say this is above a background or not. For a lot of the other contaminants, it's so patchy, even with, in our case, over 2,300 samples, really you can't interpolate a surface very well. So we're identifying a cell as being contaminated if one of the samples within it has a contamination, is above background. And of course, there are areas which we didn't sample, so we're kind of limited in some sense, even with our extensive sampling of what we actually know. And then what we'll do is I'll show you a summary map in a second. Okay. My computer is frozen. I apologize. I do not know what's going on here. Oh, there we go. I go back. Yep, there it goes. So there's simply a map of the 2,300 soil samples we collected, and they're a subset of all of them that we have collected over the years. If we look at infrastructure, which we're defining as buildings, pipelines, roads, and fuel tanks, you can see the maps on the right show the individual elements, where the total map shows a cumulative of those. And you can see that a lot of cells have all four, or a number of cells have all four of those infrastructure elements present within that 25-meter cell. Similarly, we can look at contaminants. So the dark purple area is where we have mapped either TPH or lead within each of those cells. And the yellows to reds are those other 17 elements that are much more patchy in nature. And you can really see at McMurdo, the footprint of chemical contamination is largely defined by lead and total petroleum hydrocarbons. And finally, what you can do is you can then sum all of those impacts up to start to look at which areas are impacted by multiple impacts and which may only have one or two. And so you can see from this map, and I'll show you in my last slide, some total numbers, but most of the area has only less than typically 5 impacts in each particular cell. And if we put it all together in my remaining minute or so here, the total area of impact, the total study area is a little bit less than 2.5 million square meters. 92% of that we believe was physically impacted in one way, shape, or form. About 53% had some level of contamination, and 23% had some infrastructure. And then if you look at the map on the lower right, you can see that 37% of the area had no impacts, but at 62% had one or more impacts in each particular cell, and you can see how it drops off. So by the time you get to 5 impacts, there's only about 10% of the landscape. So this approach gives us some idea to really try to quantify which areas around the station are impacted in one or more ways. And again, just a few concluding marks. So cartographic overlay does appear to provide a viable method for examining the spatial footprints around a well-studied station like McMurdo. Again, the patchy nature of chemical contamination and our even extensive but yet incomplete sampling remains a real limitation to being able to make comparable maps for these contaminants. And also what we're looking at here is just what's above background. We haven't looked at, well, how do we quantify the magnitude of the impact, as well as if we look at multiple impacts, which of these is most important? We have GIS techniques that are well established that we can use to weight these. But what I would really like some feedback on from the community is which of these impacts is most important when we try to do some sort of combined assessment. So I'm done, and thank you very much.
Thanks so much, Andrew. That was a fabulous talk and impressive coverage of that area. In terms of data.
Sorry to interrupt, but no time for questions for that one, just slightly over, so we may need to move on and hold questions for Andrew's talk to the end, perhaps.
Thanks, Jane. I've put in a question there for you, Andrew, but we can talk about that after, I hope. All right, I'd like to introduce our next presenter, Dr. Dan Wilkins, and colleagues here at the Australian Antarctic program. So go ahead, Dan.
Thanks, Kath. Just can confirm that people can hear me. Give me a hands up if you can, Kath.
Yep, all good.
Yep, and you can see my screen.
Yes, perfect.
Excellent. All right. Thanks to Andrew for his talk. That's a great precursor to my talk. It means I'll have to explain a few less things. So we're moving from the McMurdo scale down to Casey Station. Casey Station is sort of about 1/10 the size of McMurdo. So we're looking at about 120 people in summer and about 20 people in winter, but still a relatively large station by Antarctic standards. So we've done a lot of work at Casey Station, both characterising and remediating fuel spill contamination. And today I'm just going to talk about one particular site where we're using drones and a soil gas survey at one site to look at characterising that. So it's the work of a lot of people in my team. I'm presenting today, and I'm in the environmental stewardship program here at the Antarctic Division. So we're going to be talking about a site called the Cuff site, or the Casey Upper Fuel Farm, and that was the site of a diesel spill in 2018. And I'll talk a bit about how we characterise that using photogrammetry with drones, and also using passive soil gas survey as a proxy for fuel impact at that site. And we did that by deploying a grid of drill holes or boreholes and vapor cartridges, which actually sat at the bottom of those, just above the soil surface, and measured the vapor in that footprint. And then how we combine those two things to get a picture of what's happening at the site, and get an idea of what we need to do to manage that for a good environmental outcome. Okay, so hopefully these videos work all right. We're approaching Casey Station here from Law Dome over in the east, and you should be able to see the Casey buildings appearing. It's a dispersed site, a bit of an old-fashioned model for stations, lots of separate buildings. And in the green area there is the site we're going to be talking about today, the Casey Upper Fuel Farm. As you can see, this image is taken when there's lots of snow cover around. It can look quite different in the middle of summer. This is what the site looks like from the air. The spill point was at the corner of that fuel line there. It happened during a fuel transfer when a gasket gave way, and there was about 3,500 litres of special Antarctic blend diesel that came out before the pumping could be stopped. And what you can see there is the recovery effort underway. Now we'll take a look at that site using a video. Once again, that corner there is where the fuel spill occurred. There's the diesel storage tanks. You can see a large area of cleared snow up there, and underneath that we've actually got a meltwater diversion trench to try and get meltwater away. There's an Antarctic specially protected area nearby, but that's up gradient and upwind of the site. And our main receptor of concern is obviously the marine environment. And we also have Tala Valley down at the bottom of the slope there, which was the site of quite a large cleanup effort by the Antarctic program. The area of highlight in red is the Casey Upper Fuel Farm, and that's the area we're interested in characterising. As you can see, there's a lot of snow and ice. This is a photo from the recovery team that went, the recovery effort that went on at Casey Station in September. So very cold conditions. Everyone on station out there cleaning up, and they recovered about 1,000 litres of fuel, pumping that directly from the site, which meant we had about 2,500 litres of fuel that exited and went under the snow and ice. And that's where our team came in and tried to characterise where that fuel had gone. So these days we have some amazing tools. We don't have to just rely upon single aerial photographs or satellite images. We have drones available, consumer level drones that are incredibly capable. So things such as the Phantom 4, we happen to use Phantom 4 RTKs. And you know, the GPS networks are now good enough that you can use automated mapping missions, and there's very little manual control required. The main thing we need is ground control points, or networks of ground control points that are well surveyed, particularly for sites that are covered by snow and ice, so that you can go back time and time again and get things to line up. We tend to do our photogrammetric modeling in Pix4D or Esri Site Scan, and then we export that to GIS software for site management. And this is what a sort of typical flight program would look like. This is one particular flight at the Cuff, 17 minutes, the drone was in the air. Combine that over 2 different areas with a crosshatch survey, and we've got about 35 minutes of flight times, 455 photos, which we can turn into an accurate site model with a ground sampling distance or pixel size of about 2 centimetres. So talking about very high resolution here. and that's over an area of about 10 hectares. So what does that look like? So this is what the cuff site looks like. Here we have our orthomosaic draped over our digital surface model. You can see a few interesting things. So you can see over here, if you can see my pointer, there's a patch of aqua or jade, which is where we have a melt pool. We have quite a deep roadway cut through the site. And you can also see a lot of apparently relatively featureless snow or ice. And so the challenge is seeing what's going on underneath that. And that's where we decided to try a different technique to what we normally do. Often it's about soil or water sampling, but here we're talking about using vapor sampling. So we're talking about deploying vapor cartridges. They're like a sticky fly trap that pick up the hydrocarbons that are volatile. It was challenging for the team to deploy these. So they deployed 51 of these in the first season, and we only recovered 40 in initial time frame, which is about a 14-day deployment. The team back here also did a bunch of lab testing, looking at how these vapor cartridges responded to fuels we know well. The advantage of using something like this means we can tackle a site like the Casey Upper Fuel Farm, where we have all this snow and ice, with a new method. It's also very, very sensitive, and we're bringing back samples that aren't water or soil, so there's minimal biosecurity concerns. And what we used was a Beacon Environmental in the US for the provision of these and the analysis of them. So let's take a look at what the site looks like. Here we've got an overlay of multiple levels of information. So up in the top, we have our fuel spill with our hydrocarbon intensity, or TRH, modeled with contours. Then those orange pipes you can see sticking through the snow, where we drilled down and deployed our vapor cartridges. Underneath in grey and black is our modeled bedrock surface. So we interpolated the bedrock surface based upon our drill holes, plus a bunch of transects we took as well. What you can see now is the hydrocarbon intensity, or TRH signal, measured by the vapor cartridges. And you can see we've got two sort of distinct, or three distinct areas of impacts, with one over on the left side corresponding to the deeper part of the valley, the darker colours being the deeper bedrock, and one over on the red in some of the shallower area near the initial spill site. So this is what the chromatograms look like from our vapor cartridges. If you're used to looking at chromatograms for hydrocarbons, you'll notice we're sort of looking at the front end here. So we're talking about the volatiles, so we're picking up those F1 and start of F2 fraction. And they look quite different depending upon whether you're looking at petrol, special Antarctic blend diesel, or standard diesel. Now we'll go and take a look at what we actually saw in the ground at 4 different locations: B7, A11, C12, and E4. And the chromatograms are shown there. I'm not going to go into too much detail. I'm just going to point out that they all look quite different. And they showed some quite interesting things. So we have an old fuel spill over in this area, which looks like an old diesel spill, probably associated with some drums. Over here, we have something that looks like an old solvent spill. It didn't match any of our fuels that we use on station, and the best match that they could get from the reference library was shellite, or white gas, which we actually use in fuel stoves on site. So what does that mean for our site? Well, it means that when we're looking at fuel transport from this particular spill event, get this to play, we've actually got several different things happening. So over on the right, we have our relic fuel spills, which aren't as mobile. And over on the left of the screen, we have the special Antarctic blend diesel. Now it makes sense when you look at the paths of meltwater shown in blue, and also where the melt pools are. So we've actually got a correlation between the deepest part of the site in terms of the thickest snow and ice, and also these pipes or tunnels traveling through the snow, and that's moving the fuel down through the site. And in the case of the impacts here, we've just picked it up on the edge of the grid, so we don't actually have it defined at its furthest extent, but we're talking about a distance of 56 meters that's traveled from the original spill site. Not not sure how I'm going for time, so I'm moving through this quite fast because I was going over time before. So what have I presented? So we use drone photogrammetry at most of our sites now, so it's simple to use. It provides excellent resolution in 3 dimensions.
And you're at 10 minutes, just letting you know you're at 10
lovely.
minutes now.
Thank you. And we can do, thanks, Jane. And we can do it multiple times through the season. So we have what we call 4-dimensional sort of site characterization. The vapor cartridges did an excellent job. It's a new technique for us. We could discriminate fuel types and we could do that at very low concentrations. The only caveat is there's no direct correlation to concentration in soil or water samples. Anyway, that gives you a bit of a flavour for the type of work we do. Sorry it's so short, but I'm happy to take questions or talk more about it later. All right, thank you.
Thanks so much, Dan. I think in the interests of keeping to time, we'll save any questions for you till the end of the session, if that's okay. People feel free to add it to the chat or chime in at the end. All right, thanks, Dan. Our next speaker today is, I hope I get this pronunciation correct, is Dr. Abhishek Chauhan from Amity University in India. So please go ahead.
So good morning everyone, and I'm delighted to see earlier presentation and presenting at this platform. So I'm sharing my screen. Just a second. So my work is on basically fecal and total coliform bacteria in Antarctic environment. So we have participated in 34th and 35th expedition, wherein we have studied occurrence of coliform and fecal coliform bacteria in lake water samples collected from, can you see my screen? Is my screen visible?
Yes.
You should go to presentation mode.
Okay, so I will make it in a,
Yeah.
Perfect.
Good, thank you.
Yeah, thank you. So as you know, the Antarctica in the lake water samples collected from Lasman Hills and over East Antarctica. So we all know that Antarctica is a continent of extremes. It's highest, driest, coldest, windiest, and emptiest. And these are the features which everyone knows about: low temperature, having different pH levels, low organic nutrient and water availability, strong winds, and UV radiation. So these are the scientific station and tourism activities. In India, we have Bharti and Maitri station, and this glacier map is showing Antarctic. The grey ones are the glaciers, and the white and blue one is the ice. So we have taken samples from Brookness, Fisher, Solomon Island, McLeod, Sandra Rock, Easter Island, Bradman, and Stones Peninsula. So these are our sampling sites. We have taken samples, grab samples were also taken. So these are the different samples taken, and we established the sample analysis facility at the station itself. So there are different kinds of bacteria, fungi, and algae, but we evaluated coliform and fecal coliform in lake water samples. So these are the sampling locations from Lasman Hills. These are the yellow dots are showing different sampling stations, and then it's a island. It's different Fisher Island. So there were different stations are there. So in the samples, only yeah. So in most of the samples, the growth was not observed. And in some of the samples, any microorganism was not detected. Isolation of psychrophilic bacteria, E. coli, Salmonella, and Pseudomonas. So in most of the samples, again, all these were absent, but only in ST2 site, E. coli, Pseudomonas species were present. The samples were very carefully analyzed for the presence, and we did the gram staining, catalyst test, oxidase test, and also different kind of tests were done. So in that we got in Pseudomonas isolate positive, 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 and most of the samples, and at growth at 42 degree only it was found negative. And for this Pseudomonas aeruginosa, gram negative rods were there, and then positive, positive. Only in the last type of growth conditions it was found negative. So these are the different kinds of isolates. So in the present study, we assessed that environmental impact of the sewage markers such as coliform and fecal coliform bacteria and are finding fecal were absent in all different lakes, Bruckner's and Bruckner's Peninsula, East Antarctica, while coliform bacteria were present in 2 lakes, P2 lake and P3 lake of Bruckner's Peninsula, and 9 lakes, L1C, NG, L1D, NG. These are the different sampling names we have kept of Bruckner's Peninsula. The presence of coliform bacteria in Antarctic lakes may be due to the human population, all the anthropogenic activities which we are doing, which is living at the research station. Also, maybe it is because of other bird droppings and other things. Regular monitoring at research station is required to control the release of microbiological contaminants, and these contaminants can cause further several diseases in humans and other animal species. Research on the presence of fecal coliform bacteria in Antarctic lake water could be helpful to provide a better estimate of the human impact. Thank you so much. So any question, I would be happy to answer.
Thank you for that presentation. We're just checking that chat to see if there are questions coming through. We have some time. We've got a few minutes, about 5 minutes.
I would like to say that on the further studies, we have submitted this abstract, but on the further studies, we have found some actinomycetes with antibacterial activity. So now we are trying to explore those actinomycetes species for the drug development. So we will be doing standard, antibiotic test against that. And recently we have been working with the group on development of a drug using those actinomycetes species as an antibiotic. So this is the next step which we are doing. And regarding this, that fecal and coliform bacteria, I think more rigorous studies must be done and more samples has to be taken on the trend analysis of their presence in Antarctica. So that also we have proposed further.
I had a question. I'm not sure whether I've missed this in your coverage, but did you have any way of identifying whether the E. coli was from like an animal source, like a bird source, or human?
Yeah, that we could not assess, but I think we will find out the methodology. If the source can be assessed, then the remedial measures can be taken. So as I've mentioned, it may be anthropogenic, it may be a bird source. So that study needs to be carried out.
Mm-hmm. Thank you. We have a couple of other questions and comments from Dan Wilkins. It's excellent. Thanks, Tanu. There seems to be a lot of potential for actinomycetes with antibiotics. Can you comment on that at all?
What is it?
There seems to be lots of potential for actinomycetes with antibiotics.
Yes. So So as I said that we have tested the actinomycetes received from the samples. We have tested them against standard antibiotics, and we have found its potential as a antibacterial. So we are further exploring it now for the drug development.
Thanks.
I have a bit of a question, just we've got a bit of time. I'm not sure if our next speaker is with us either. Tanu, sorry that I introduced you incorrectly there as well, my apologies. My question was really relating to how much work has been done on the transfer of this bacteria to the native biota. Can you comment on that? You know, what is the risk of this to native biota, whether we're talking wildlife or smaller organisms?
Obviously, but the plus point is that, you know, because of the very cold environment in Antarctica, the multiplication rate is very less. So the risk may be less, but nonetheless, we need to really analyze, you know, further impacts of their presence, which is alarming, because you know, anything because as the global warming, climate change is happening, so there are chances that their multiplication rate becomes high and their concentration may build up. So I would like to do further studies in the lakes because the studies were carried out 4 years back, and so I think there is a urgent need to analyze other different lake samples to counter confirm the findings, and also the current concentration or current presence of these coliform bacteria. So they may have adverse impact because it depends on their presence, how much is the concentration.
So no, I know that it's not this situation is not unique, and I suspect that anyone who's looked for fecal coliform bacteria in wastewater discharges at stations around Antarctica will find it. And it is alarming, not just as a normal contaminant, but we're really talking in the realm of introduced species here and risk to our biodiversity in Antarctica. So it's you know, as Antarctic Treaty signatory nations or and people that are preventing the introduction of other species, it's interesting that the focus hasn't been targeted to microbial and bacterial communities, which are actually have much more likelihood of invading and taking hold in Antarctica. So I really appreciate that talk for highlighting this as an issue.
Thank you. And I would like to collaborate with anyone who would be interested in such studies, either on the drug development and also on further analysis in the water samples or snow samples. Snow samples, it's not much, but water samples, lake water samples are very important to analyze to know the current situation.
All right, we are due for our next speaker, but I've heard from the IT team that we haven't seen Dr. Carlos Banchon in the within the participant list. So if you are there, Carlos, or someone is here to represent you today, could you please speak up? Because I haven't got a withdrawn of your of this presentation, but we can't seem to find you within the list of participants. Anyone know of Carlos or what might be going on there? Just leave for a couple of minutes. I have had some messages from people saying, do we have to turn up for the whole session? They had time clashes, and I've been messaging no, just make sure you're there for the talk. Carlos wasn't one of these, but we'll just give it a couple of minutes to see if he comes online or a colleague comes online. In the meantime, if we want to revisit any questions for previous speakers, by all means, certainly there was a question that I put up for Andrew around the biological receptors in the environment, whether they had been measured, and Andrew's answered as we often find, we're funded to look at biological responses. So often, we're looking at the chemistry and not always the biology, purely because of funding and the difficulty in working with biological communities. So thank you for that answer, Andrew. See if there's anything else.
I had a question for Andrew, if there's a little bit of time allows. Just wondering, Andrew, whether you might have covered it in the methods, and apologies if I didn't grasp it, but did all the sampling happen over, is it a single snapshot, or did it happen over a number of years or seasons, the collection of that data?
So we've collected data from 1999 to 2015, missing a couple years. What was presented here was 2003 to 2013, where the sampling design was the same.
Yeah. Okay. Great. It's a nice data set.
Yeah, fabulous coverage of that whole area, Andrew. That's right.
Yeah. Nice data set and a nice design. Yeah.
I take it no one's commented whether they know of Dr. Carlos and whether he might be coming or not. And there's no colleagues online from Spain, are there, that might shed some light on that? We've got about 8 minutes till the next speaker, if that's the case. So I'm loath to suggest people go and have a tea break because they might not come back, but this probably is a good opportunity to go grab a cup of tea or a glass of water if you'd like, and the rest of us can stay online and keep having a chat, or yeah. So the next talk will be at in about 7 minutes time. So please feel free to drop out if you need to go and have a break, and then please come back.
Dan, if you're there, we have a question from Andrew. Or maybe we might have to come back to that again when everyone's back on at their desks.
Sorry, Andrew, I've just got back. Haven't been at my desk, so I'm seeing your question now. Yes. So the short answer is we can't correlate a vapor concentration trapped on our cartridge directly to a soil concentration. At least not for this site. So we do have another site that I wanted to talk about but didn't have time. So we have an old diesel spill at Casey from the 1980s, and we did actually a bit of a comparison there between soil samples, TRH in soil, and what we measured trapped on our vapor cartridges. So I didn't get a chance to talk about that. The difficult thing about the cuff site that I talked about is that it's very difficult to get samples from it. So we're drilling down through 4 or 5, or in some cases even 6 meters of snow and ice. And it's difficult to know where the fuel is traveling. So we do know that we have little sort of melt pools and sort of melt tunnels through the ice, and they can be pathways for the contaminants to travel. So it's not an easy answer, but we have very few techniques available until the daylight's at the bottom of the slope, basically 200 meters away. So the vapor cartridge is a way for us to start to see where it is on the slope and to work out how quickly it's traveling. But yeah, it's not an easy one-to-one. Hope that answers your question.
Thanks, it does. Pretty cool, actually.
Yeah, we were surprised by this, and we were also surprised, as I say, by the legacy site, where we were actually getting decent vapor hits from a fuel spill that happened, for almost 40 years ago. So it is quite amazing to see.
All right, so I think we've got to move on. We're now back on to schedule, which is great. So I'd like to introduce our next speaker, which is also one of the co-conveners of this session, Dr. Catherine Brown. And here she is. Thank you.
Thank you. Have you got that screen up there for the slideshow?
Yes, the screen is up, Kath.
Right. So the research I'm presenting today on contaminant risk assessment in Antarctica using microinvertebrate toxicity tests is being done together with colleagues at the Australian Antarctic Division. So in Antarctica, ice-free land covers less than 1% of the total area. And these terrestrial ecosystems are the most important areas biologically. And this is also where most human activity occurs around scientific research stations. Unfortunately, terrestrial sites, as you've heard, have been contaminated, mostly at these research stations. And although we have improved our environmental practices, there are still the risk of accidental fuel spills and leaching of contaminants from past pollution events. So to assess impacts from contaminated sites, environmental toxicology has an important role. This involves toxicity testing with native organisms, together with understanding the chemistry of the contaminated sites and the local biodiversity. However, there are few protocols for toxicity tests with Antarctic species, especially for terrestrial systems, and sensitivity data is limited. One of the challenges we do have is the low biodiversity in Antarctic soils where there are no macrofauna. So we have microinvertebrates such as rotifers, tardigrades, and nematodes as our potential invertebrate test species. One Antarctic species we've been working with is Plectus marii, which is a common and ecologically important nematode, and it's found within moist soils and mosses. We've developed culturing techniques and toxicity test protocols based on its life cycle and its reproductive strategy. This nematode has a life history traits that are commonly seen in other Antarctic invertebrates, most notably a very long life cycle. This is in contrast to standard test species from temperate regions. We trialled a number of endpoints from egg hatching success to adult survival. We found survival of J2 juveniles was the most suitable endpoint. The J2s are easily sourced from cultures and they're often most sensitive to contaminants. So we've developed a standardized toxicity test using this stage. Our test uses aqueous exposure in sealed vials or well plates, usually with 4 replicates for each treatment and 10 nematodes per replicate. So here are some results from toxicity tests with copper. And this figure shows survival data for J2 juveniles up to 28 days of exposure. So you can see nematodes responded to increasing copper concentrations, but there was a minimum of 21 days was needed for models to be able to estimate sensitivity. And this is around 6 times longer than tests with temperate nematodes. From the response data, we generated sensitivity estimates, shown here with 95% confidence intervals. The LC50 values are copper concentrations estimated to cause 50% mortality. LC10 values for 10% mortality are commonly used in guideline derivation as an acceptable level of risk, having no long-term effect on populations. So we see after 21 days exposure, the LC50 is 478 micrograms of copper per litre. Sensitivity increases with exposure time, and at 28 days, we see the same lethal effect from 117 micrograms per litre. A similar trend was seen for LC10 estimates, with a 28-day estimate of 30 micrograms per litre. And if you'd like to see more on the test development and copper toxicity, here's a link to a paper published in Environmental Toxicology and Chemistry. So, as I mentioned earlier, and as you've seen in previous talks, diesel fuel spills are some of the most common pollution events in Antarctica. Now, diesels are a complex mixture, and the chemistry of a fuel spill also changes with aging, including through loss of volatile compounds and through biodegradation, which breaks down hydrocarbons into polar metabolite compounds. So to help inform risk assessment and remediation at Antarctic fuel spill sites, we ran experiments to measure the chemistry and assess toxicity of Antarctic diesel as it aged in soil. In this lab experiment, we spiked clean Casey soil with special Antarctic blend diesel. Samples of the spiked soil were aged at 3 degrees for 45 weeks, which represents multiple years of Antarctic summer soil temperatures, to see how toxicity might change with aging. At multiple times during aging, samples were removed and then stored frozen to be used for toxicity tests. And then we made elutriates from freshly spiked and aged soils to use as the exposure medium. Now, elutriates are made by washing soil with water. They represent the soluble and so the bioavailable part of toxicants in soil. Elutriates can also represent contaminated water that flows from a spill site during the Antarctic summer melt season. So to measure the chemistry of the spiked soil and elutriates, we used a suite of analyses. Concentrations are reported as total petroleum hydrocarbon, or TPH, which includes hydrocarbons and polar compounds, and as TPH after silica gel cleanup that removes the polar compounds and so reports just the hydrocarbons. TPH can be further divided into fractions based on carbon number ranges to better understand the composition. The concentrations of a number of aromatic hydrocarbon compounds were also measured. We tested the toxicity of elutriates made from soil at 10 aging periods, and we made treatments by diluting the full strength elutriate. We estimated lethal effect concentrations, and for tests where an LC50 could not be estimated, ANOVA was used to identify the lowest observed effect concentration. So the results of the chemical analysis are a little complex, so I'll just highlight some key points. The concentrations and composition of what we start with is quite different from what we end with. We see the losses from soil during the aging process and an increase in proportions of polar metabolites, shown in blue, as hydrocarbons biodegrade. The lighter aromatic hydrocarbons are rapidly lost from the soil. Concentrations in elutriate are more than 1,000 times less than in soil, and composition's different. Elutriates have more of the F3 and 4 fractions compared to soils, and they're dominated by polar compounds, which are more soluble than most hydrocarbons. We estimated the sensitivity of nematodes to TPH in elutriates and to the hydrocarbon-only component. So elutriates of freshly spiked soil were the most toxic, and aged fuel was less toxic, and the steepest decline in toxicity was seen in the first 6 weeks of aging. So we couldn't model an LC50 for every test, but effects were still evident in all tests, including for the most aged soil, and this is despite hydrocarbon loss from soil and an elutriate almost entirely composed of polar metabolites. So looking at how toxicity relates to the chemistry, there was some evidence that nematode sensitivity was related to the proportion of the F2 fraction of TPH in elutriates. And this F2 fraction contains the acutely toxic and more soluble lighter aromatic hydrocarbons such as the naphthalines. So the next step is to use this nematode to directly test the toxicity of soils from fuel spills that are being remediated, such as in biopiles at Casey Station, which you can see here. This will help support decisions around soil remediation and reuse. So we've estimated critical effect concentrations for this native nematode to some pollutants of concern in Antarctica. These findings contribute to predicting ecological risk at existing fuel spill sites in Antarctica, to deriving site-specific remediation targets, and to environmental guidelines to assess ecosystem health. We're continuing with development of other species as toxicity test organisms, including rotifers and tardigrades, so we can include estimates for more taxa when assessing risk from contaminants to Antarctic terrestrial ecosystem. Thanks for your time, and I look forward to any questions here or further contact by email.
Thanks so much, Kath. We've got a bit of time for some questions. Our next speaker's in 2 minutes, and that's a recording. So any questions for Kath? Kath, I shouldn't really ask a question, being co-author on that, but anyway, it's a question that I think was posted earlier from someone. I think it was Dan actually had mentioned availability of species, and he said, will it be challenging to sample enough terrestrial species? So I just wanted you to comment maybe on the availability of test species to work with in Antarctica versus, say, Macquarie Island or the Antarctic Peninsula.
Yes, as I said, we as far as fauna is concerned that live within the soil, it's basically the microinvertebrates. So that creates a challenge just in itself in working with micro creatures, and we don't understand a lot around the life history and ecology of many of the species, and there's many that are still being identified. So having more species gives us a greater certainty around when we're coming up with sensitivity estimates. If we combine from multiple species, then we can get estimate that's more representative of the species. So having this nematode cultures is a good first step in being able to have a standardized test. So working on developing with things like rotifers and tardigrades, if we can develop protocols and get more understanding of the the creatures, then we can combine more into having more robust estimates.
Great, thanks, Kath. I think we should move on just to keep on schedule. So our next speaker is Clara Pathos, and I believe our IT team are going to preload that recording for us. Hopefully.
Good morning, dear audience. Today I'm going to talk about the research project entitled Activity of Acinetobacter johnsoni in the Degradation of Low-Density Polyethylene. The use of polyethylene is a global problem that has affected the environment. However, this accumulation has begun to be seen notably affecting firstly aquatic species as they die from starvation or strangulation, and human health because when has throughout food chain, indirectly microplastic, since the main diet for people is composed of seafood. For this reason, the research is carried out to evaluate the degradation of polyethylene using a extremophile microorganism. For this purpose, Antarctic microorganism isolated from Pedro Vicente Maldonado station was used. A pure culture was obtained from the sample, which was set in Luria Bertana broth and kept at a temperature of 15 grados Celsius. Due to the taxonomy complex, a molecular techniques were applied. First, DNA extraction using master pure kit. Modifications were made in the quantities of the established reagents. Universal primers, 27 forward and 518 reverse, were applied in the polymerase chains reaction. The results were visualized in electrophoresis help. The polymerase chains reaction products was purified using a Kiogen PCR kit. Finally, the samples were sent to sequencing. The result was analysis in silico using Genius Prime and NCBI BLAST program. The samples employed with primer showed a band with molecular weight of 1,500 pairs pass, indicating that the amplification of 16 genes. Through a liquid program, it was confirmed that the microorganism belonged to the species Acinetobacter johnsoni, being the first report in Antarctica. This species has an open path, a great adaptability to different environments. It can be well distributed in natural environments under cold ecosystem conditions, and can also be found in clinical settings. Antarctic microorganisms were used because they have shown encouraging results in bioremediation. If the environment conditions like pH, temperature are improved, the capacity of bacteria could be improved. Furthermore, Acinetobacter johnsoni has already been studied in other studies to degrade nitrogenous compounds and oils. Being a heterotrophic species, it can depend to wide variety of carbon source. The plastic used was oxo-biodegradable low-density polyethylene, which ultraviolet light and twin 80 pretreatments were applied. The degradation requires physical, chemical, and biological process because the pretreatments helps to despolymerize the plastic with the bacteria act with their metabolism to transform oxidized products into carbon dioxide and biomass. Twin 80 has a surfactant effect that causes flexibility in the polymer. The surfactant, due to its ethylene oxide composition, allows it to act on oxidation when it comes into contact with the plastic, filling the space free in the polymer. Twin 80 is a low toxicity compound, so it has been used in bioremediation. In the case of the ultraviolet light, this allows the activation of the pro-oxidants additives present in the plastic, which cause the polymer rupture to accelerate. In addition, it's a friendly process with the environment as long as the radiation is not so high. The first consists of a spur of 365 nanometers, and the second, a twin 80 solution was prepared, and the plastic was washed in the solution with the help of a mini shaker. For the gradation development, a bioreactor batch was designed with 2 compartments. In the first compartment, a gross kinetics could be measured, and the second compartment contained sodium hydroxide solution. Through this compartment, it was possible to calculate the amount of carbon dioxide released. This design was used to prevent the escape of carbon dioxide into the environment, which can be harmful. Therefore, a sodium hydroxide solution was placed in the second compartment to capture the gas. This reaction caused carbon dioxide to increase as hydroxyl ions are consumed, forming sodium bicarbonate as result. Additionally, infrared spectroscopy and dry weight analysis were performed to demonstrate the physical change of the polymer. As a result, it was obtained that the plastic with twin 80 allowed the bacteria was obtained the best dynamic of the ground, and a reduction in the weight of polymer of approximately 1 gram was visualized. However, the carbon dioxide was low. It is displayed in the infrared spectroscopy, where there was not major change in absorbance and transmittance spectra. In this way, it was determined that although the capacity of Acinetobacter johnsoni was low, what stands out is capacity of adaptability in the medium under stress conditions. Besides, it is the first report of the polyethylene degradation process, which opened a path of future research. In conclusion, only one sustainable alternative is offered to solve a part of plastic problem that continues to be challenging in science. Thank you.
Okay, I don't think we can really ask any questions of Clara since she's not with us, but we've got 25 minutes until we commence session 2 of solutions to pollution. So maybe if we have, let's see if there's any interest in generating some conversation now for the next 5 minutes, see where that goes. And if not, I invite you to take a break and then rejoin us in 23 minutes for the next session. But myself and the other 2 co-conveners will stay online and see if there are people interested in chatting. If you are, I suggest either use the chat or raise your hand, and then we can unmute you and have that open discussion. So we'll stay online for a bit and see if people are interested in doing that. Also, if you have any questions that haven't been asked of previous speakers, please feel free to either put that in the chats, in the chat line, we'll monitor that, or or raise your hand, virtual hand, I should say.
I have a question for Andrew. You have in the chat that used a microtox test for marine species, and can you explain a little bit more about what samples you tested and what you found?
So we basically used microtox to test marine sediments in area. So we kind of did a sediment quality triad approach. And it was kind of what you expected. About the only thing that we found was a lot of our control sites were also toxic, probably because of spicules. I'm blanking. It's midnight here. So they had some high spicule contents because it was below spicule mat. So it was kind of not as indicative as we would like. I hope that answers your question.
Yes, I imagine you might have had different sediment characteristics too at particular sites besides the spicule variation.
Yeah, there were slightly. I mean, the control sites were just a few hundred meters away from the other sites at McMurdo, so there was some change. There were some differences in the sediment sizes, but not a lot. But areas where we had high concentrations outside of the areas we had sponge spicules, we did see higher toxicities.
And I also had another question around your TPH. Did you do any comparison of whether did you highlight the aged and the fresh spills as far as areas that were the high levels of contamination? Did you sort of track those?
No, we did not. So this was kind of truly a monitoring program that was not a basic science approach. It was looking at trying to monitor McMurdo's footprint. So when we integrated the chromatographs, we really did just do TPH. We didn't break it down for this study into the different components. So we didn't really track age, if that makes any sense, to look what was fresh and what was not fresh. It was just what was there.
Yeah.
All those things would be wonderful to do.
Yes. Yeah, no, it's a really comprehensive spread, and yes, like you say, there's only so much you can do on a broad scale, particularly.
I think it'd be a very complicated story as well if you break it down.
Yes.
Well, it's cumulative impacts, right? I mean,
yep.
For most bases where you're doing the activity has been the same place you've been doing the activity for a long time, which is a good thing, but it creates a very complicated signal, as you said.
Yeah. Sorry, we've kept you up so late, Andrew. That's a particularly bad time slot for you.
It's okay. I'm afraid I'm going to skip the next session.
Fair
but
enough.
I'll be with you tomorrow.
Yeah, fair enough. That's very understandable. I'm just tracking through the questions again. I don't think there was anything else that hadn't been addressed. But if there is from any participants, please just raise your hand.
I think we come back to all of them that are in the chat.
What was that? Sorry, Kat?
I think we've come back to all the questions that were in the chat at some point.
Right. Yeah. I reckon we'll have a break, but I do encourage those of you who it isn't close to midnight to come back. And also, and just a further reminder of the poster, the e-posters, they are available on the platform. Anytime you want to go in and check them out, you can download them, print them out, read them at your leisure if that's what suits you. And hopefully after tomorrow's session, the talks tomorrow, we might have a bit of time for any presenters of those e-posters that are keen to have a bit of a chat about their work to do so. But yeah, please get online and have a look at those. So it's I think we've just got about just a little over 15 minutes before the next session starts. So I think let's take a well-earned break. And oh, sorry, Jane.
Oh, just to give people one more chance, I just had a note go to the Q&A to say attendees cannot post to the chat. So we need to use the Q&A section, question and answer section, rather than the chat. So just to give attendees, if you've been trying to post to the chat and you do have a question, post it to the question and answer. We'll be able to see it there. We can activate you to allow you to speak as well.
Okay, thanks, Jane. We'll switch to Q&A unless the IT team sort that out or tell us otherwise. Yep. All right, we'll see you back here in 15 minutes for those of us who can make it. See you. Thank you. And thank you very much to all those speakers for that session. Wonderful job. Great diversity. All right, I think we are just about on time now. Can I just have Kath and Jane online, just confirm you can hear me okay?
Yes, all good.
Great, okay. Good morning or good afternoon or evening, everyone from across the globe, from wherever you're joining us. And thank you for coming to this session. I welcome you to this second session on solutions to pollution, contaminant impacts and remediation in Antarctica. I'd like to, I see that many participants have stayed online from the last session, but we also have some new participants. So I'll just go through some introductions and run through some housekeeping before we get into it. My name's Dr. Catherine King. I'm a Principal Research Scientist at the Australian Antarctic Division, and I'll be chairing this session. I'd also like to introduce my colleagues, Drs. Catherine Brown and Jane Wasley, who I've worked with at the division as co-conveners. And a shout out also to the IT team on the ground who are assisting in delivering this session. Some housekeeping. So each speaker has been allocated 10 minutes, followed by 2 minutes of questions. So speakers, please keep track of your time and we will let you know if you're running over time so you can wrap up. Please ask your questions by typing into the Q&A box. So the last session we had some people that weren't able to see the chat box. So if you can put your questions into that into that function, we will keep track of that and speakers can answer there as well. So the Q&A, please. Everyone except the current presenter and convener should be muted at all times, except for the which should happen automatically, except for the discussion at the end of session, which we'll encourage you to all participate. We've got about 20 minutes at the end of the session. And you hopefully will be aware that the presentations are being recorded. They'll be available via the SCAR virtual platform in the past event sections at the conclusion of this session, and also via the SCAR YouTube channel, so within 24 hours of the session end. So if you don't want your talk to be published on the platform, please let us know at the start of your talk and the IT will pause recording. And similarly, if you don't want your presentation to be made available via YouTube, send an email to opt out to the SCAR organising team. I think that's it for housekeeping. And we have, just to let you know, we have 8 presentations in this second session. This is the second of 3 sessions on solutions to pollution. The third and final session will be tomorrow with quite a few talks on microplastics, 2 hours earlier than this session. So that's tomorrow if you want to join in. I think that's just the other thing I wanted to mention was e-posters. So as you know, there've been e-posters also submitted to this conference, and that exhibition space is available on the event platform. It's up and running. You can browse through the posters at any time for the whole duration of the conference, and you can also comment on them in that space. And hopefully after the end of tomorrow's, the third session of Solutions to Pollution, we'll have a block of time for discussions, and any people that have submitted an e-poster that want to run through those with participants, we can do that at the end of tomorrow's session. I think there's about 11 or 12 posters for this session for you to check out. I think that's all I need to mention at this stage. So yeah, just put your questions in the Q&A function and we'll keep track of that. So let's kick off the session with our first speaker. So Zani Dharam, I'd like to, are you there? So are you there?
Yep, I'm here.
Okay, wonderful.
Yeah, but I regret to say that somehow my camera is unable to be open today. I'm not sure why.
That's okay. Can you share the screen? Can you share?
All right, so I'll share my screen.
Perfect.
I cannot start my screen sharing while the other participant is sharing. So, okay, so
Please try now.
right, can everyone see my slide?
Yes, perfect. Thank you.
All right, so a very good morning, greetings to everyone.
Full screen, please.
Full screen. Yep. Thank you for reminding. Right, greetings everyone. My name is Shazani. So for today's presentation, I'm going to present a small part of my PhD project that is being supervised under my supervisor, Dr. Siti Aklimah Ahmad, and it is on statistical optimization and kinetic studies of molybdenum reduction using a cyclotolerant Antarctic marine bacterium. So jumping on to my introduction, there has been many pollutants that has been reported in the past few decades in the Antarctic, especially heavy metals, and it has been shown to negatively affect the ecosystem. And then for my project is mainly on focusing on heavy metal contamination, and these are some of the papers that have reported on the contamination of heavy metals in the Antarctic ecosystem. It has been found, heavy metals has been found in sediments, seawater, as well as the soil of the Antarctica. So there are some sources of heavy metal pollutants, which is mainly from the human activities or the anthropogenic activity, or from the natural source. So the main source is from the human activity, from local as well as the foreign source. So from foreign is mainly from nearby countries in which these contaminants are being transported by the long range transport. And then for local, it's mainly from human activities such as the research stations as well as the tourism industry. So my focus is mainly on molybdenum. Molybdenum, because it has been reported in the past few decades, the presence is rather snowballing, and it has become one of the concerns in the Antarctic. And a brief introduction of molybdenum. Molybdenum is a rare metal with various oxidation states, and it is biologically important as it is one of the components for several metalloenzymes. And its properties are ductile, resistant to corrosion, and has high melting point. So because of these properties, molybdenum has wide industrial importance. It has been used in many industrial products such as in paints, and it has been used in production of alloys and machinery components. So although molybdenum is biologically important, being exposed to a prolonged high concentration of molybdenum can cause toxic effects. For human, it has been reported that it can cause hallucination, hyperuricosuria, seizures, as well as aching joints. While in animal studies, in cattle and sheep, it has been reported to induce copper deficiencies, which lead to lameness and osteoporosis. While in rodents, it can cause many symptoms such as growth retardation, as well as reproductive failures. So one of the solutions to solve the pollution problem is through bioremediation. Bioremediation is basically the exploitation of organisms such as plants, fungi, and bacteria, in which their metabolic potential can degrade or immobilize hazardous pollutants. Pollutants which include both organic and inorganic to an ecologically safe level. For the bioremediation of molybdenum, especially in Antarctic region, there has been only one report or one research has been done, which is using the Antarctic soil bacteria by Ahmad in year 2013. And for bioremediation of molybdenum, it is using the reduction pathway in which the molybdenum is being reduced by an enzyme called phosphomolybdate reductase to a compound, MO-blue. MO-blue is basically the end target, in which it is a colloidal compound that can be filtered out from the environment. It has a vivid blue color that can be measured using spectrophotometer, using at 865 nanometer wavelength. So for my research objective is first to optimize the molybdenum reduction using statistical approach RSM, and the second one is to ascertain the kinetic profile of the molybdate reduction. For the method is for the first one, the optimization using RSM. It is done using the CCD, or Central Composite Design, in which it is used to determine the optimal values of conditions selected and to study the interaction between significant factors. The software design expert has generated 86 runs for my study. And for the kinetic, I have selected 4 different types of models to study the relationship of the reduction rate as well as and the substrate concentration, which is the molybdate concentration. And the curve fitting is being done using the Curve Expert Professional software. The workflow is rather simple, which is we grow the bacteria in a specific media called the low phosphate media, and the molybdenum blue production is being observed with a spectrophotometer at a wavelength of 865 nanometer. So the bacteria used for my study was the Marinomonas sp strain AQ5A9. It is an Antarctic marine bacteria isolated near the vicinity of Base General Bernardo O'Higgins reclamation station in Antarctica. It is halotolerant as well as cyclotolerant. It is gram-negative, motile, aerobic, and is widely distributed in marine environment, including in polar regions. So for the results and the discussion from my first objective, which is the optimization, we did the optimization for 6 different factors, which are the salinity, temperature, carbon, nitrogen, and molybdenate concentration, as well as the pH of the media. And then the response is the optical density for the molybdenum blue measured by spectrophotometer. This is the result ANOVA table for the RSM, in which the model as well as the lack of fit shows that the model is very significant. And the R square is above 0.85, which means that the model and the CCD is acceptable and it can be used for further application. So for the models, it has for the probability value less than 0.05 shows significant parameters. And the most important part is the interactions between 2 factors, in which it's being highlighted in the red box. There are 8 significant interactions that has shown to be highly significant. And then it is these parameter interactions are being generated. The surface plot of these interactions are being generated. This is important to identify the peak or the maximum value for each parameter, in which is being tabulated in this table. Sorry, it's not comparison, but it's more on the optimization table. So these values have been is the result from the optimization using the RSM, and the incubation period is being shortened down to 4 days, and the molybdenum production is at 2.2 OD. So for the second result and discussion for my second objective is to ascertain the kinetic profile of the molybdenate reduction. This graph shows the curve fitting for the 4 different models to the experimental data indicated by the red black circles. Further details are being tabulated in the form in this table, in which we measure the value for RMSE, R square, and AICC value, as well as the value of the coefficients. So from this table, we can see that IBA model has the best fit mathematically as well as marginally compared to the other 3 models. As we can see that the RMSE value as well as the AICC value are amongst the lowest, and the R square value are amongst the highest between all 4 models.
You're at 10 minutes. Just
all right. So let me conclude the presentation. It's basically conditions for molybdenum reduction must be optimized to ensure successful remediation, as well as the refining the optimal conditions using statistical response surface methodology is ideal. And last but not least, the IBA model giving marginally the best fit for molybdenate reduction compared to the other models. That's all from me. Thank you.
Great, thank you for that presentation. I think we have just a minute. If there's any questions, if you'd like to put those in the Q&A. It's Izani.
Yep. Okay.
I don't see any open questions at this point, but I just had one for you. I noticed that with the optimum salinity was quite high compared to what
Yes.
would be normal in a marine environment.
Mhm.
Could you comment anything around that and what it means in a practical sense?
Okay, so basically the bacteria is isolated from marine environment, and it is highly tolerant towards saline environment. But because these bacteria at first is being adapted in from the start is being adapted in high salinity condition, so it can actually survive in a very saline condition, as well as also in a very low saline condition up until 30, down until 3.5. The value is of 35 ppt, sorry. So it doesn't make much any different because previously we did the one factor at the time. So the optimization does not really provide much difference when it comes to wide range of adapting the bacteria in wide range of saline condition. So in a practical sense, I do think that the bacteria can also reduce molybdenum in a normal saline condition, which is the Antarctic salinity level, which is around 3.5%, right? All right. So thank you for the question.
Thank you.
All right, we best leave that there. We can have further questions at the end of the session. So I'd like to introduce our next speaker to you, Paula Rivas Alvarez. Hopefully I've said that right, and Paula is here. Okay, this was not withdrawn, so just give it a couple of minutes in case there's some issues, or if anyone knows of Paula and whether someone else might be presenting on her behalf that isn't being admitted.
Paula's name is not in the panelists list.
Oh, I can see that, yeah.
Yeah.
Are there any co-authors of Paula's here that know what's going on there? Because
there's a message in the chat, Kath from the SCAR IT team, saying they have a recording from Valentina in place of Paula.
Oh, beautiful. Thank you.
Can you play that recording?
Microbial communities. Can we play it?
Please go ahead and play the recording if it's available.
Okay, we're doing it.
Thanks, Dan.
Everyone, my name is Valentina Carrasco, and I'll be presenting our work, Soil Microbial Communities Capable of Biodegrading Polycyclic Aromatic Hydrocarbons in the Presence of Heavy Metals. The Antarctic continent constitutes one of the more unique environments on the planet, which has given rise to great interest in exploring and investigating it by the scientific community. However, in recent decades, the anthropogenic input has been increasing, especially in regions with a high density of bases, due in part to the fact that the main source of energy for the operation of the bases and transportation is petroleum-derived fuels. This has become an environmental problem due to contents spilled of fuels such as diesel, which is very harmful and when it's spilled into the environment, mainly due to the harmfulness of the hydrocarbons that compose it. Bioremediation with bacteria is a low-cost and low environmental impact alternative that allows decontamination of soil and that, under the provisions of the Antarctic Treaty, requires the use of endogenous microorganisms. On the other hand, diesel contains significant concentrations of some heavy metals that affect the efficiency of biodegradation. However, its effect has been scarcely studied to date in Antarctica. The objectives of this work were to select microorganisms from Antarctic soil samples capable of biodegrading hydrocarbons in the presence of metals, and to analyze the bacterial communities before and after the selection process. 2 soil samples from diesel, cadmium, and lead contaminated sites were collected from Pratt Base and Escudero Base, South Shetland Islets, and also 2 samples from non-contaminated soils around the same sites were collected as controls. For the first objective, we realized a selective enrichment in batch cultures at 28 and 4 degrees Celsius for 5 and 8 weeks, respectively. As carbon source, we use glucose, diesel, phenanthrene, and also phenanthrene with cadmium or lead. During the process, we measure microbial growth and perform a qualitative monitoring of degradation as culture medium turns from colorless to yellow due to the generation of this metabolic intermediate. It was found that both at 28 and 4 degrees Celsius, growth in diesel and phenanthrene occurred in the cultures that were initially inoculated with contaminated soil samples, indicated in red. In these same cultures, it is where biodegradation associated with the color change in culture medium was observed. This strongly suggests that there is a microbial selection of microorganisms that are capable of biodegrading, and that they also do it in the presence of metals such as lead and cadmium. The soil microbial communities were analyzed through 16x amplicon sequencing before and after the selection process. Before the selection, we found that in the 4 soil samples appeared these bacterial genera, which are cycrotolerant bacteria that have been previously reported in Antarctic soils. When analyzing the 2 soil samples of contaminated soils, we found an arrangement of genera that biodegrade hydrocarbons, indicated in green. Among them, the genus Acanindiges stands out, which has reported strains that can only use hydrocarbon as carbon source. It is noteworthy that these samples were also contaminated with heavy metals, and in this regard, the genera indicated in blue have been associated with resistance to metal and have been found in soils with high levels of them. We also analyzed the communities after the selection process. In diesel, we mostly found Pseudomonas, which is one of the most reported genera associated with the biodegradation of hydrocarbons such as alkanes and aromatics. And these other genera also appear in cultures inoculated with the contaminated soil samples, where we also observe bacterial growth, and these have also been associated with the biodegradation of hydrocarbons. When using phenanthrene as carbon source, this other genera was selected, of which those indicated in green correspond to bacteria that biodegraded phenanthrene and also other aromatic hydrocarbons. Regarding the temperature, we can see a significant increase in Polaromonas at 4 degrees Celsius in this sample, and this genus has been reported mostly in cold areas, which could explain this phenomenon. In the presence of metals, we found a favoring of the biota genus, and it has been reported in this genus that low concentrations of cadmium favor the biodegradation of phenol, which is a petroleum derivative. We can also see a favoring of Sphingobium genus in the presence of lead, and this is a genus that has been strongly reported as resistant to this metal. In conclusion, these results reinforce the importance of studying the effect of co-contamination of hydrocarbons with metal on microbial communities in areas as sensitive as Antarctica, and also the relevance of considering heavy metals when selecting the optimal microorganisms for an efficient bioremediation. To finish, I just want to invite you to visit our e-poster of a work related to this topic. Thank you.
Okay, how are we doing for time? Well, no questions, obviously, with that pre-recorded talk. We'll just hold off for a minute or so for the next speakers, just so we remain on time in case there's any late people joining. A reminder that if you have any questions for any of the speakers today, please use the Q&A function within Zoom, and myself and the co-chairs will be making sure we keep track of that.
Kath, we're bang on time to start next
time now.
Yep.
Okay.
27. Yep.
All right.
Just alert you to a couple of messages around changing in presenters' names, and that the fourth presenter is still as an audience. So I've passed that message on to raise their hand to be added as a presenter.
Great. Yeah, I've gone through that list, but thank you. Yep, if the IT team can make that happen, that'd be wonderful. Thanks, Kath. All right, so I would like to introduce our next speakers. This is a bit of a double act here, which is lovely. Good example of international collaboration in this space. So we have with us for this presentation Greg Hintz and Lucas Alvarez. So please go ahead. Welcome.
Just share that screen here. And hopefully that's working.
Yes, we have that, Greg. Go ahead.
Great. So so this, as you mentioned, this is a bit of a double act. This is Lucas and myself presenting from both Australia and Argentina. And both Argentina and Australia have spent quite a number of years managing and remediating fuel contaminated sites in Antarctica, applying practical remediation strategies from studies to pilot scale to full scale remediation. And obviously there's quite a bit of variability across Antarctica, either from the types of fuel that different countries use to site conditions. And there's a need to understand the similarities and differences in order that we take advantage of the learning and benefits of an international collaborative approach. This work includes, of course, the input from many people across multiple institutions, and the logos that are presented on this screen are just a small sample of that. My name is Greg Hintz. I'm from the Australian Antarctic Division, and I'll be presenting with Lucas Martinez Alvarez from the Argentinian Antarctic Institute. Like most stations, Australia and Argentina have contaminated sites. Both of us have historic and contemporary fuel contamination that we're responding to, and this results from events such as fuel transfer and handling, failing infrastructure, or human error. Occasionally from shipping accidents, although they're less common. And we do have legacy contamination from either fuel dumps or waste dump sites. And for this talk, there's a focus on fuel and hydrocarbon remediation, particularly in the terrestrial and the nearshore environment. Human activity occurs mostly in the coastal ice-free zones around Antarctica, which also happen to be biodiversity hotspots. And it's imperative we protect and treat the impacts in these areas. Australia's Casey Station and Argentina's Carlini Station are at direct opposite sides of the continent, and it creates an opportunity to study these diverse locations and develop approaches that are sensitive to local conditions. This includes how we make decisions, evaluate risk, apply treatment strategies, and monitor sites. And Argentina and Australia have taken the opportunity to compare, contrast, evaluate, and share our different approaches. In the talk today, we want to compare or consider some of the differences, which includes how we test assumptions, because what works well at one site might not work well at another. It allows us to test different parameters and confirm what parameters matter. We look at enhanced experimentation where we can test alternatives, and in doing so, we can prove the approaches prove the approaches we take and test that they're robust and repeatable. Today, we look at differences in fuels, site and operational conditions, and differences in soil and soil communities. And all of this learning contributes to the cleanup manual as part of the CEP, where we can consolidate our learning and the approaches that we take the differences into account that are useful across the continent. And I thought I'd hand over to Lucas to talk about some of the specific differences and similarities that we see across our sites.
Hello, good day, everybody. Can you hear me properly?
Yes.
Great. So thank you, Greg. My name is Lucas Martinez, and when we focus on the fuel types on both sides of Antarctica, or different programs, both Argentinian and Australian, happen to have different diesels with different properties and different chemical compositions, as you may see in the graph. This means that, as you know, hydrocarbons are a complex mixture. That means that may have different evaporation degrees, different biodegradation processes and rates, and also different toxicity. And when we studied our diesels, we found, for example, that Argentinian Antarctic diesel is a little bit lighter, has a little more amount of light alkanes, but as well a little more polycyclic aromatic hydrocarbons were detected, which means that toxicity may be different for both of our fuels, which means also that the health of the soil after bioremediation may be different from both our remediation programs, and we need to focus on that as well when we think in a bioremediation scheme. Regarding the climate and the environmental conditions on both our sides of Antarctica, there are a lot of differences as well when you program to perform a bioremediation strategy. At Carlini Station in the Antarctic Peninsula, we have currently a lot of heat, sadly, due to climate change probably. Mean temperatures are going high, and we even have a lot of rain, a lot of rainy days with temperatures above zero. When compared to Casey Station, we can see that the mean maximum temperature at Casey Station is way lower, which may be affecting the biological activity of our remediation programs. And those things are important things to take in consideration when planning to perform bioremediation schemes in Antarctica per se. These operational issues are not only temperature-like things, but also the limited access that we have to our stations, which is limited mostly to summer, make us try to find new ways to perform effective bioremediation programs, probably limited to summer months because of the temperatures allowing biological activity, which means that we have to try different strategies as reactive barriers or different water treatment systems, as you see in the top, in the bottom left picture, or try to biopile soil inside geomembranes, which perform some kind of protection of the soil from freezing, and trying to keep temperatures a little bit higher than freezing point. And these strategies are the only way that we can perform bioremediation schemes in Antarctica, and effective bioremediation schemes in Antarctica for large fuel spills, and are the only way that the Antarctic system we found for our programs to take cost-effective and low-maintenance programs for recovery of soil contamination. These methods, although that may be a little bit long in time, are robust and are reliable, and also depend on the different conditions that soil may have on our sites. Carlini soils and Casey soils are basically very different from a very sandy soil in Carlini and a more silt-like soil in Casey. This means that these differences in soil structure and conditions may affect the mobility of the hydrocarbons, may affect the treatment per se, and we also need to think about the nutrients concentration and the rocks that may be included in soils. Also, the soil biodiversity in both soils are going to be different, but the main genera or the main key players in hydrocarbon degrading may be the same. For example, in both our cases, in both our biopiles, we found that Rhodococcus was one of the key players in the remediation process, but also Arthrobacter, for example, in Casey, or Pseudomonas in Carlini. So the diversity of the soil may be different in both sides of Antarctica, but the key players may be related or may be the same when thinking about our bioremediation process, which make us think that it's probable or at least feasible for doing it in any part of Antarctica. Regarding nutrient amendment, we also optimized the nitrogen and phosphorus ratios, and we reached an optimum at different levels for both sides on Casey and on Carlini, but following a similar process, testing which is the best ratio, of which is the best nutrient amendment to optimize the effective bioremediation of the soil. Always have to consider a lot of different aspects in this optimization, adjusting different kind of nutrients, of trying different nutrients, measuring their effectiveness, whether they are organic or inorganic. Well, all those kind of things are implicated in the optimization, but at the end we found that they are very different from what is considered as a reference for nutrient amendment. And finally, for the measurement of the hydrocarbons, we also use different techniques. For example, infrared spectrophotography.
Excuse me, Lucas, you're
yes.
at 10 minutes, just letting you know you need to wrap up. Thank you.
Okay. And from the perspective of the Australian Antarctic Division, also using gas chromatography. Back to Greg.
Thank you. So we considered a range of similarities and differences, and essentially proved that we can remediate sites in Antarctica. And most importantly, what we're looking for here is to continue this multinational approach, and where possible, invite other nations to join in this multinational approach to cleaning up Antarctica and protecting the Antarctic environment. Thank you.
Great, thanks so much, Greg and Lucas. Really good example of collaboration there and some great results, so appreciate that. I think we probably need to move on with time to our next speaker, but reminder that at the end of this session we do have a block of time around 20 minutes if people want to hang online to ask any questions at that point. So our next speaker is Zheng Suen Lim, who hopefully is online and able to share their screen. If you can unshare, Greg, that'd be great. And Suen, we have seen that request as well. So IT team, if you can please acknowledge that as well. I'll request for no recording, please. Okay, please commence, Suen.
They were online, but they seem to have disappeared again now.
Okay. Yeah, they were in the panelist. No,
Yeah.
no, Suen's still there. So let's just give a couple of minutes. Hopefully she'll come back online.
Hi, I'm so sorry to inform you. Something was wrong with Shan's Zoom. So is it okay if we just postpone her session for a bit until she sort it out?
Are you proposing to switch your talks? Is that what you're saying?
Yeah, just carry forward with mine first.
Yeah, I think anyone who's here is probably here for the session, so that's a great solution. Okay,
Yeah.
so let's proceed then with Razidni. I hope I'm saying that right. Razidni Wong. So welcome and go ahead, please.
All right, so I'll share my screen. Okay, so I hope everyone can see my screen.
Yep, that's great. Perfect.
All right, so greetings everyone. So I will be presenting my findings today on the statistical optimization of Antarctic bacterial consortium as diesel degrading agent at low temperature. So moving to the introduction. So as I'm sure everyone is familiar here, Antarctic is the Earth's most southern and most pristine region. Though it seems to be uninhabitable, the human activity in Antarctica has actually increased over the year according to the IATO data in 2019. So most of human activities in Antarctica is powered by diesel combustion. So that leads to a very significant fuel consumption in some of the Antarctic research stations. And undeniably, most of the Antarctic research stations nowadays have been working on renewable energy. So this is just a very vague amount of annual fuel consumption before the use of renewable energy. So Antarctica is protected by the Antarctic Treaty System, and in 1991, the Madrid Protocol was established, where Antarctica is designated as a natural reserve devoted to peace and science. So a few annexes that are worth taking note for the purpose of my research is that all human activities in Antarctica after the Madrid Protocol requires an environmental impact assessment, and procedures for environmental monitoring must be put in place before it can be conducted in the area. And the most important one is the prohibition of intentional introduction of non-native species in the area. So the problem with Antarctic environmental protection is that though the regulation is there, up until today, the interpretation of the protocol is it's more to a self-interpret thing. So next, different countries or different operators might have different understanding or standards of this regulation on the Antarctic environment. So it might not be a complete protection of the environment. So other than that, very obviously, very limited availability of machinery and facilities, other than the fact that it's also logistically challenging and inappropriate. So as human activities keep on increasing and non-viable remediation, this leads to diesel pollution in Antarctica. So usually it's sourced from two sources, which is mentioned just now. Number one is accidental leaks, and this is a paper in 2019 that reported a leak of 4,000 liters of SAB in Casey Station, which is unfortunately concealed for more than 3 months due to the weather and snow cover. And other than that, though very rare, as Lucas mentioned just now, boating accidents also occur in the region, and this is just a very brief summary of how the accident impact the environment. So a lot of researchers have focused on the use of single species bacteria, indigenous bacteria, to degrade diesel in the region. So as I've mentioned just now, as a foreign microorganism or foreign organism in general is prohibited from entering the region, so the use of indigenous bacteria is really important in this topic of research. So it is said that conglomeration of several bacterial species can enhance the bioremediation approach if the species have synergistic effect. So, which brings us to the objective of my research, which is to use Antarctic Bacteria Consortium to, as a diesel degrader, and this will be optimized by a statistical approach, OFAT, and also RSM responsive-based methodology. So for OFAT, one factor at a time, 6 parameters are chosen: salinity, nitrogen source, and concentration, pH, temperature, and initial diesel concentration. So as for bacterial growth throughout the experiment, it will be measured through a spectrophotometer, and as for degradation efficiency, it will be done through gravimetric analysis. So then the data will be carried forward to a secondary optimization using Plackett-Berman design and central composite design, where Plackett-Berman will mostly focus to screen the significant factors to be carried forward to CCD, which can measure the relationship of these factors. So the results that I wanted to share is, as of now, Bacteria Consortium BS9, the one I'm using, it works best at 0% salt, and the preferred nitrogen source is ammonium chloride at 1 gram per liter, and it also works best at a neutral pH 7. So as for temperature, it works best at 10 degrees Celsius, not surprisingly. But also what's interesting is that even at 20 degrees Celsius, it can also degrade up to a certain amount, which is not much difference from 10 degrees Celsius. So as for diesel concentration, the best concentration, degradation efficiency is recorded at 3% of diesel. So these, the highest and second highest parameters are carried forward to Plackett-Berman design, where it will have a low and high experimental value. So according to this design, the best degradation efficiency occurs at 4% diesel at 15 degrees Celsius at a neutral pH with no salt and 1.5 gram of nitrogen concentration. And this is the ANOVA table for Plackett-Berman design. The model is significant with an R-square of 0.932, which deems the data to fit to the model. And the significant factors from Plackett-Berman design is salinity, diesel concentration, and temperature. And these are the 3 factors that will be carried forward to central composite design, where the algorithm will give a central value, a low and high value, and also 2 extreme values. So the findings of after central composite design is that the consortium actually works best at the central point, which is at 0.25% of salt at 12.5 degrees Celsius with a diesel concentration of 3%. So all 3 highest degradation efficiency occurs at the midpoint. So this is the ANOVA table. Again, the model is significant. The lack of fit is not significant with an R-square of 0.956. So the significant interaction between factors can be found between salinity and diesel concentration, as well as temperature and diesel concentration, where the p-value is less than 0.05. So this is the umbrella plot for the central composite design. As you can see here, the best degradation occurs somewhere between 3.5 to 4% of diesel with a salinity of around 0.4 to 0.5. And as for here, the best diesel concentration, the best degradation is again at 3.5 to 4% of diesel with a temperature of around 13 to 15 degrees Celsius. So this is after the identification of second order effect of these factors. So according to the algorithm and the math that is done through this software, the best factors occurs at 0.35% of salt at 13.38 degrees Celsius with a diesel concentration of 3.66%, where the degradation can be found at 69.037.
But for the sake of a continuity of the research, the diesel concentration is up until 4% with a salinity of 0.4%. So the favored degradation is deemed to be at 67%. So this is a validation for the design, as the p-value is more than 0.05. There is no significant difference between the predicted and experimental value. So the model is applicable and also reproducible. So the conclusion is that OFAC and RSM can be used as a good tool to optimize diesel degradation. So as you can see, the comparison between pre-optimized and post-optimized, where the diesel concentration is up until 4%, and incubation period is increased by 1 day, and degradation efficiency does seem a bit lower than before, but keep in mind that this is 4% of diesel concentration. So bacterial Consortium BS9 can be a practical agent for diesel remediation in Antarctica. So these are my references, and thank you everyone.
Thank you, Razini, for that presentation. I'm just not able to see whether Zheng has been able to get online. We're really running over time now, so if she is, that would be good to start her, but I can't see access there within the panelists.
No, just very briefly. So there might be some IT issues or.
Yep.
Yeah.
Okay, she's back in. I've just promoted to panelist, so let's see whether Zheng comes on. I don't know if there's anything the IT team can do on the ground to help facilitate this. Have we got the IT team in India there to help out with this? We have Sue and Lim as a presenter trying to be uploaded as a panelist, but that doesn't seem to be working.
I think CT is saying something, but you're muted, CT.
Actually, my students still waiting in the waiting room. Xuan.
Yeah, I have promoted her to a panelist, but it doesn't seem to be working. So
thank you.
Unless the IT team can do something, City, can you just check that again for us, that she's still listed in attendees?
Yeah.
If not, can I proceed first? Because Xuan is texting me if it's okay if I proceed first.
Is it okay?
Yeah. Okay, I just want to get this sorted. I don't know if the IT team can work on this in the meantime. That would be great.
Okay, okay.
Right.
Let me just share.
Thank you so much. So our next speaker then has become Nadira Zakaria. Please go ahead and you put that in presentation mode. Beautiful. Thank you.
Okay, so hello everyone. My name is Nadira Zakaria, and I'm here to talk about my 14-page presentation titled Diesel Food for Antarctic Surface Seawater Bacteria. So Antarctica carries a huge prominence into understanding how this world came to be. It is remote, harsh, and largely uninhabited. Despite it, this continent and the Southern Ocean are central to the Earth system, where changes in Antarctica have implications for the rest of the planet as climatic and oceanic changes. So Antarctica is also a key observing platform in understanding how ecosystems adapt and respond to environmental changes and the direct impacts of human activity. Now, human activities in Antarctica are largely dependent on fuel as an energy source for heating, and most of the activities they carry out at research stations. So electrical power is generally provided by diesel generators. So these fuels are shipped in from nearby continents and stored in storage tanks at the research station. So these past few decades have seen a rise in how important Antarctica is and how it plays a role in science, economics, and geopolitics. Henceforth, the need for petroleum transportation, storage, and consumption was expected with the increase of human activities to set up research stations, tourism, and aircraft, all the while under strict regulations of the Protocol on Environmental Protection to the Antarctic Treaty. So Antarctica's nearshore environment is generally ice covered for much of the year, resulting in low energy sea conditions. Because of this, oil spills are at risk of becoming encapsulated in Antarctic ice for many months. Low temperatures also causes biodegradation rates, slower attenuation to be much slower than in temperate areas. So the consequences for oil spills in Antarctica are potentially more critical than elsewhere. Therefore, in situ biological remediation techniques are increasingly considered the best approach in Antarctica, specifically in targeting fuel contamination, which is limited in the summer months. So for my project, seawater samples were collected over the course of 2 expeditions to Antarctica, specifically at Base Bernardo O'Higgins in the Antarctic Peninsula. Samples were collectively screened for diesel degrading abilities before proceeding to the optimization of growth and diesel degradation, using 1 factor at a time and proceeding to response service method. So based on the highest percentage of diesel removal after a period of 7 days, sample O2B, which can be seen in the red box, was selected, originating from uncontaminated surface seawater within a 3-kilometer radius of said research station. So here we have the growth conditions, which were optimized in an attempt to enhance the diesel degrading efficacy of the selected bacterial consortium. In graph A and B, you can see that diesel degradation was highest in pH 8 of phosphate buffer and at 3% salinity. So diesel degradation here can be seen highest to be at 10 degrees Celsius, where all these 3 physical conditions all agree with the conditions of seawater in Antarctica, where the alkalinity and salinity agree with the growth conditions of psychotrophic bacteria, because it is able to survive up to 25 degrees, as can be shown in graph C, even though diesel degradation is not promising. So in graph D, we can see that diesel degradation was also highest, where ammonium nitrate was applied, and at a concentration as low as 0.5 grams per liter, diesel degradation was still applicable. Here in graph F, we can see that increasing concentration of diesel supports bacterial growth in proportion to the amount of diesel being used as a carbon source. However, we can see at 4.5% and higher, the degradation starts to decrease and finally is inhibited at 5.5. So after narrowing down to significant parameters, the optimization proceeded to using RSM to finding the optimized conditions. It was revealed that there were 3 significant relationships between pH, salinity, nitrogen concentration, and temperature, and also diesel concentration. So firstly, we have graph A, which is between salinity and nitrogen concentration, where decreasing levels of salinity would require higher levels of nitrogen. Secondly, at lower temperatures, requires high levels of nitrogen to sustain efficient diesel degradation. Third and lastly, diesel degradation was indeed stimulated to higher levels when temperatures remained low at 10 degrees. The concluded conditions is that diesel degradation was optimal at 2.5% salinity, 12 degrees, 1.5 grams per liters of ammonium nitrate altogether. So the time points identification of the chosen bacterial consortium was chosen at day 0 before diesel treatment, day 3, and day 6 after 3.5 diesel treatment. So day 3 represents the exponential phase, while day 6 is the final day of the incubation period after optimization. You can see that using packed biosequencing, we can see that there are shifts in the community composition in response to the high diesel concentration. we can see that at day 0, proteobacteria, mainly consisting of the Cyclobacter species, genus, represents almost 97%, in contrast to Planococcus from the Firmicutes. At day 3 of diesel treatment, Pseudomonas slowly began to replace Cyclobacter, finally decreasing at day 6. In contrast, we can see that the Firmicutes evidently increased from 0 to day 6, where the dominating genus was Cyanobacterium. So attention has also been given to heavy metal resistant bacteria in improving hydrocarbon bioremediation success, which highlights the importance of the synergistic interactions between these different bacterial species in improving biodegradation success. So surface seawater have lower concentrations of heavy metals than in soil and sediment samples. Here, 11 types of heavy metals at 0.5 ppm were used. We can see that, diesel degradation was inhibited by silver and mercury. We know that both silver and mercury have no known biological functions in any cell. So to conclude today's presentation, we're looking at naturally occurring marine bacteria, microorganisms found in non-contaminated surface seawater, which has the potential to utilize diesel as a carbon source and can be further studied into becoming a tool for future bioremediation efforts in Antarctica. Thank you very much.
Great, thank you for that presentation. Appreciate all the shifting that was done there between you guys. That was wonderful, and I think we now have Sue and Lim online. And before that becomes not a reality, I would like to invite you to give your talk, please. So Zheng, Sue and Lim, if you're ready to go, that would be great.
Hi, yes, sorry for my technical issue right now, and I'm also still facing a problem to open my camera, so I'll just present as usual. I hope it doesn't affect. Yes, would you please start recording, because this is unpublished data, so that would be great if you could start recording. Thank you very much.
Put that request through. Thank you.
All right, thank you. Can you see my screen?
Yes.
All right, good afternoon, everyone from Malaysia. My name is Zhang Xuan, and I'll be presenting on the taxonomy identification and phytoremediation mechanisms of an Antarctic microalgae acting on diesel hydrocarbons. So phytoremediation is an enhanced bioremediation approach which employs plants, fungus, and algae to mitigate pollutants. As my colleagues have presented on the bioremediation using microbial, and for me, I'll be using algae as my remedial agents. And phytoremediation usually involves higher plant, which is evolved to have more complicated structures such as vascular system, roots, and leaf. However, in Antarctica, higher plant growth is not favored, and algae is abundantly found on terrestrial and marine ecosystem. Studies on phytoremediation using microalgae in the existing data mostly focus on heavy metal and other inorganic pollutants in temperate regions. Only few studies have been reported on hydrocarbon removal, and no research have been conducted on Antarctic research. Some of the very popular tropical hydrocarbon degrading microalgae species include Corella, Selenostrum, and Syndesmus. Nevertheless, microalgae show promising results in biotransforming recalcitrant hydrocarbons such as naphthalene and phenestrine. The use of microalgae is advantageous in bioremediation as they possess high surface to volume ratio, less nutritional requirement as compared to bacteria. It can also adapt to mesotrophic growth, and it has high bioabsorption capability. They are potential remedial agents for the removal of various toxicants, and it is reported that Antarctic algae demonstrated higher tolerance than microbial communities and invertebrates of the region. Diesel is a widely used fuel in Antarctica to powerful vessels, vehicle, and research stations. Different pollutants can arrive in Antarctica through long-range transportation by means of ocean and air currents, as well as from local sources, as previous researchers has already explained. So the leaking of pipeline, domestic sewage disposal, as well as oil leakage during the station refilling and grounding of vessels can contribute a big part to this issue. Diesel toxicity is a great concern. Is commonly addressed with the composition of diesel. Diesel chiefly consists of hydrocarbons with a majority with a paraffin range from carbon 9 to carbon 25, and also aromatic compound. The fuel used in Antarctica is treated differently during the refinery process to prevent jouling of diesel in cold temperature. For instance, high molecular weight paraffins are selectively reduced in diesel to lower its density and viscosity. And upon oil spill, natural attenuation removes most of the volatile hydrocarbons. Depends on the nature of soil, it can affect the mobility, toxicity, and also longevity of oils. Degradation rate can be much slower, and the oil seeps deeper into the soil and can be trapped within the unsaturated zone where the dissolved oxygen is lacking. And on the other hand, it can form emulsions in contact with seawater, which has increased its viscosity. The returning process may be reduced, and the oil may persist longer in the environment due to the extreme polar environment and freeze-thaw cycle in Antarctica. And this figure illustrates the oil-ice interaction, how the oil is encapsulated in the ice and re-released into the marine ecosystem with higher bioavailability, hence a higher toxicity. So this raised the problem statement. Some attempts have made to assess the impact of hydrocarbons on algae, but mostly from the temperate regions. So to our knowledge, there has been very few studies to address the effect of hydrocarbon pollution on polar microalgae, as well as the applicability of using Antarctic microalgae in degrading the hydrocarbon. So in this study, it aims to study the diesel hydrocarbon degradation potential of Antarctic microalgae isolated from Greenwich Island, Antarctica. The isolated strain was identified using polyphasic taxonomy approach and then characterized for its physiochemical property using basic MATH method. And the phytoremediation study was conducted to understand the mechanisms behind. This is the research study, research designed for identification and characterization, where the algae will be observed under light microscope and phase contrast to observe the morphological appearance, as well as analyze based on the ATS and ITS sequencing. The cell characterization will be conducted based on microbial adherence to hydrocarbon MATH method that is proposed by Rosenberg in 1991. The solvent used will be chloroform, ethyl acetate, and also hexadecane. For the phytoremediation study, the algae will be incubated with diesel for 9 days. And during this incubation period, gravimetric analysis using the hexane to extract the diesel will be conducted. This culture will be first be centrifuged to obtain the cell-free supernatant and pellet. The cell-free supernatant is where we will extract the residual oil in the media, and the pellet will first be extracted and then harvest the oil that is absorbed onto the algae cell, and then the pellet will be sonicated to extract the oil absorbed into the cell. So the biodegradation efficiency will be calculated based on these values, and the result will be processed using gas chromatography FID. So this is a finding. The microalgae isolated in the culture had a morphology conforming to Staphylococcus bacillus. It can form short filaments up to 4 cells in one. It has a length from 5 to about 12.8 micrometer long in a cylindrical shape. But based on our molecular analysis, based on the concatenated 18S and ITS sequences, the algae show the highest similarity to genus Staphylococcus, a lineage that is newly evolved from the redefined Staphylococcus clade. And in a recent published, the Antarctic Stylococcus taxa, including the Tritostylcoccus, was found to possess strong resistance towards the antibiotic cyclohexamide. And the findings even interested more because we are curious to know about what this Antarctic microalgae can do with its resilience. So from the MITH analysis, the physiological properties of the cell wall, such as the degree of hydrophobicity and surface charge of the cells, was studied. The adhesion of chloroform and ethyl acetate was compared.
Okay, my same with Xuan Cat. We don't want to post in the YouTube, okay? Because some data is still unpublished. Okay, so I want to.
All right, let's just try that again. Let me share the screen first before I start it up.
Hi, my name is Bill. Here in this presentation, I will talk about my research of assessing the toxicity of copper, cadmium and lead to the Antarctic terrestrial nematode Plectus marii. This work was done with colleagues from the CSIRO, the Australian Antarctic Division and the University of Wollongong. Often when thinking of Antarctica, we picture a pristine wilderness of ice. And for the most part, that's correct. However, there is significant legacy contamination of old stations and abandoned waste sites from a time before strict environmental regulations were put in place, as highlighted. Invertebrates can survive. Because of this convergence of human impact and biodiversity, there is a great need to assess these impacts on the local ecosystems. So why are we doing this work? Firstly, the Antarctic Treaty, to which Australia is a signatory of, specifies the need to clean up and prevent environmental impacts. Secondly, there is a need for environmental guidelines to inform current and future remediation works and to help assess environmental impacts of future developments. And thirdly, given the difficulty of collecting and culturing Antarctic organisms, there are only a few published studies with robust toxicity data on terrestrial Antarctic species, and only one published using terrestrial microinvertebrates. The test species for this study is the Antarctic nematode Plectus marii. Nematodes are commonly referred to as roundworms and can be found all around the world. This species is endemic to Antarctica, predominantly existing in soils around the continental margin. As with many cold climate species, they have a much slower growth rate and longer lifespan compared to their temperate counterparts. Additionally, given aqueous conditions only occur during the Antarctic summer melt, Plectus marii undergoes annual freezing and desiccation and can remain desiccated for long periods of time. The life stage used for this study was the J2 juvenile, which is the first stage following hatching. We conducted a series of metal exposures to the nematode at different concentrations to see the effect of these concentrations over time. To set up our bioassay, the first step was to create a pore water matrix that was representative of the pore waters found in the soils of Antarctica. To do this, clean Antarctic soils underwent rotary extraction for 24 hours with millipede to simulate the summer melt. These were then vacuum extracted. The next step was to spike in our metals to create our concentration series. For testing, we used culture well plates with a 1 ml volume. 10 J2 juveniles were transferred from the cultures into the 1 mil well on day 0 and fed with E. coli. The nematode mobility was then assessed periodically throughout the 28-day exposure using a microscope. Our meta-analysis was done using ICP-AES. Nematode mobility was assessed periodically over 28 days, and the sensitivity to the metals increased with exposure time. A trend of delayed response to metals for polar species relative to their temperate species is commonly seen. Delayed response is often attributed to physiological characteristics for polar species, such as slow metabolic rates and long life cycles. Chemical reactions and kinetics are also slowed under lower temperatures. Today, I'll be discussing the results of the 28-day endpoint. Using the 28-day data from the previous slide, we can plot dose response curves with the metal concentrations on the x-axis and the percentage of mobile organisms relative to the control on the y-axis. Here we see a clear relationship. As the concentrations of the metal increase, the mobility of the nematodes decrease. Models fitted to the data can be used to calculate concentrations causing a 10 and 50% effect on mobility, which are referred to as the EC10s and EC50s. Here we see that copper is by far the most toxic of the 3 metals tested, with an EC10 and EC50 value of 46 and 182 micrograms per litre. These copper results compare well with Brown et al. 2019, where an EC10 and EC50 value of less than 47 and 117 micrograms respectively were recorded. Cadmium and lead were found to be of similar toxicity, with cadmium being slightly more toxic. The results of the study suggest that Plectus muriae is relatively tolerant to lead and cadmium and sensitive to copper. Interestingly, the same metal-specific tolerances were observed in other Antarctic metal toxicity studies, with a microalga showing similar trends. The EC10 values for all 3 metals far exceed the Australian guideline values, as well as generally exceed concentrations reported in soils and pool waters near contaminated sites in Antarctica. While these values are comparatively high, it is still possible that Plectus muriae and other soil invertebrates may be exposed to concentrations well in excess of values tested in the study, particularly in areas adjacent to contaminated sites. In a previous study by Snape et al. In 2001, they showed concentrations of copper and lead well in excess of 1,000 and 6,000 micrograms per litre, respectively, in pool waters around the old Casey Station tip site. Summer meltwaters could potentially mobilise these metals as a concentrated metal pulse into interstitial waters within soils in the surrounding areas, likely elevating the concentrations well above the EC10 values recorded in this study. While there is only one previous study assessing copper toxicity with Plectus muriae, a considerable amount of toxicity testing has been done on other nematode species, namely C. elegans, a temperate nematode. Comparisons of metal toxicity with other nematode species is difficult, given Plectus muriae inhabits unique environments and extreme temperatures. Temperate nematodes live for only several weeks though, a much shorter lifespan than the Antarctic species, and as such, comparison to shorter exposure times may be appropriate. Plectus muriae had somewhat similar sensitivities for copper at 28 days exposure with an EC50 of 180 micrograms per litre to that of C. elegans after a 4-day exposure of 256 micrograms per litre. However, these are some of the most sensitive estimates reported for temperate nematodes in response to copper, with many tests only lasting 24 hours with EC50 values in excess of 10,000 micrograms per litre. Interestingly, comparisons of cadmium and lead toxicity in C. elegans after a 4-day exposure show cadmium and lead to be more toxic than copper, with EC50 values of 60 micrograms per litre for both metals. This heightened sensitivity to cadmium and lead contrasts with our Antarctic nematode results. To summarise the key findings, we found that copper was more toxic than cadmium and lead. We found that previously measured soil of contaminated sites in Antarctica have exceeded the EC10 values calculated in this study. And we also found that copper toxicity was comparable to 4-day toxicity data for temperate nematodes, whereas cadmium and lead toxicity data was not comparable. Thank you very much for listening to my talk. I really hope you enjoyed it. If you have any questions or comments, please feel free to post and comment in the chat or send me an email which is shown above. Thank you.
Okay, hopefully we got there in the end and you captured most of Will's talk. So I'd just like to take this opportunity to thank all our speakers in this afternoon's session for a wonderful, diverse set of presentations and wonderful research being done around Antarctica in this space. We've got about 20 minutes up our sleeves now if there is a desire for an open discussion, or if anyone wants to post some questions specifically to any of the presenters, since we kind of didn't have time for any questions throughout the session. So I'll open up the floor to that. And if you have a question, please post that. If you want to be admitted to speak, raise your hand and we can add you to the list of panelists as well. So either of those options, Q&A or raise your hand and we'll add you in. If anyone has anything else they just want to discuss from this group of experts, that would also be welcomed. If there's any more broader questions people have about remedial technologies, remediation, the use of algae, the toxicity tests that have been presented, anything like that, this is a great opportunity to do so. So I encourage that as well.
Yeah, I just had an observation and maybe some questions around with the algal growth, the optimization in I think the 3 studies where you've optimized conditions for the algal growth and breakdown of oils and diesels. The optimum temperatures are around about 10 degrees, in fairly close to that for each of you. Do you have any insights into how this might work in a practical sense in remediating a fuel spill or and the realities of working on the ground in what might be 0 or sub-zero temperatures and any obvious challenges that are around in optimizing your growth?
Hi. So for practical purposes, because hydrocarbon degradation appears to be more efficient at, I would say, higher than normal Antarctic temperatures. So the application of enclosed environments to support biodegradation, such as incubation tanks or bioreactors, I think would be a practical measure to address this.
Yeah, yes, Kate, I want to add on. Yes, because last time I just do the biodegradation without the bioreactor, and then all my sample are freeze. It's frozen, so it cannot be degraded. It's failed.
Yes. Yes, and that puts a rapid halt to everything, hey, once it's frozen. Yeah. So as you say, bioreactors and having them within building spaces. So yeah, there's a challenge with the volume that you can process, but as you've got a fairly quick time where you've got insignificant absorbency of like 6 to 7 days in some of them, then yeah, I could see that you could work through a particular volume in say like a summer season. Yes. So yes, getting those technologies developed and refining is an ongoing kind of thing, which, as we know, with the bioremediation trials done in biopiles, it takes a lot of research and then refining of your methods and then working out the technical details. So yeah, it's great to see your work using algae as a bioremediation options.
There's a comment in the Q&A from Scott Stark. Perhaps similar setups to the biopiles at Casey for treatment of contaminated soil, I'd suggest or guess. Thanks, Scott. Yeah. Let's try that. While people are thinking if there's anything else I want to discuss, just a reminder, there's the final session tomorrow, session 3. Will be at 500 UTC time, commencing. Oh, is that right? Sorry, no, it won't be. It will be at 1 UTC time. Is that correct? I think that's right. So the same time as our first session this morning. So
3.
Like to join in that.
3, Kath. 3 UTC,
oh, 3.
I think.
3 UTC. See, I'm all over this. Sorry about that. 3 UTC. That'll be our final session on solutions to pollution. And immediately following this session tonight, there's also a session on emerging contaminants of concern. So if people are interested in that one as well, similar theme as this.
We have another comment from Scott Stark around treatment, and he suggested all supporting bacterial or algal communities on membranes under optimal conditions for contaminated water treatment. Yeah, is that using membranes? Is that something that you've given some thought to as an option as far as a technological engineering solution?
We've also considered different approaches rather than membranes to water treatment. But yes, we played around with different kinds of ideas. And membranes, we Okay, so we thought that membrane was a bit maybe further up in our development of the project, because right now we're still at our first and second stage.
Yes, this is just our first and second stage of my studies. We will add on it.
And I think that immobilized bit would be one of the very practical and interesting question to take in, as we are speaking about membrane.
Try to entrap the sample, the bacteria and also the algae in the Chelengkam bits. Still in the process, we want to try is it efficient or not.
Great, lots to look at again.
Yes.
Yeah.
Greg, have you got any comments on that from the Australian perspective and what we've been thinking about back here for water treatment in particular? Sorry to put you on the spot, but you might be. Oh, you're on mute, sorry. Can you unmute yourself? Oh, there you are.
So you mean what sort of approaches we've been considering with water treatment?
Yeah, yep.
Yeah. So our water treatment has considered, I guess, 3 main issues. One is treating water on site that's contaminated with fuel. And for a lot of our work, that's focused on the in situ permeable reactive barriers. So using capture materials like activated carbon and zeolite. For water that's contaminated, such as in biopiles or surface water that we know we've got large volumes of that we need to treat in certain ways, we've been using active pumping water treatment, particularly sediment removal and removing hydrocarbons in columns containing capture media. And more recently, we've been working to remove nutrients as well. So in the remediation process, when we add nutrients, sometimes we'll end up with excess nutrients in the water phase, particularly where that might include elevated levels of nitrite, perhaps. And we want to remove that before we end up taking the remediated soil and then doing something with it, like reusing it. In other words, the nutrients become a contaminant more so than the hydrocarbons. And so we've been using processes like ion exchange resins in columns to process large quantities of water. Obviously, keeping the systems warm is important. So we've been using modular water treatment that we've been developing in shipping containers.
So it's more on the chemical and the physical enhancement or treatment side, Greg, rather than any sort of supporting bacterial communities within the water treatment. Certainly the biopiles is a different situation.
Yeah, there has been some interest in both looking at permeable reactive barriers in how we can promote microbial activity to essentially prolong the life of a permeable barrier. And we've also been looking at the impacts that microbial or algal growth in an active pumping system might have on either prolonging the life of media or fouling media, if that becomes a practical problem rather than a part of the solution.
Right. Thank you. Got about 8 minutes left if there's anything to raise.
I have a question for Greg. It's about the permeable reactive membrane. You mentioned that backwater fouling could be an issue, and I've read that several papers that actually applies such membranes do have the issues of water fouling. So how would in a large scale, such as in Antarctica, how do we improve this? Because membrane is a good approach, but water fouling backwater has always been there. So how do we address and improve this?
I'm not sure I can answer that question quickly.
Simply put.
Yeah, I'm just thinking what we've done. With our permeable, our in situ permeable systems, biofouling doesn't seem to be an issue, particularly for water movement through those systems. So we deliberately choose particle sizes that still enable water movement and water treatment. And we often use multiple materials, so multiple stages within the barrier systems that might be nutrient release, capture media like activated carbon and zeolite. So there's multiple stages to it. So it's not just a single membrane. And we don't deliberately prevent microbial activity. In fact, where possible, we encourage it. And we, from memory, we haven't seen any issues of biofouling occurring and stopping these processes from working. In fact, temperature is the bigger issue. Having frozen ground inhibiting the movement of water to being able to treat it, and sometimes being able to actively heat a permeable reactive barrier so it continues to function when we want it to is the bigger issue.
Thanks, Greg. looking if there's more questions. That's it at the moment. All right, if there's nothing more that people want to discuss in this forum today, I think we can wrap up the two sessions for today. And I'd like to particularly thank all our presenters for doing a wonderful job. I apologise for IT issues, which were, I think, inevitable. That was my worst fear, but reasonably well. And thank you for speakers switching around to accommodate that. That's much appreciated. Don't forget the e-posters. Check those out, and if you're available to join us tomorrow for the third session in Solutions to Pollution, that would be wonderful. I think there's a real focus on microplastics tomorrow, microfibers and plastics in the environment. So if that's of interest, then come along to that at 03 UTC. All right. Thank you, everyone. Really great to see you. So many smiley faces online and from around the world. It's been a great afternoon for us and evening or morning for you guys. All right, thank you.
Thank you, Kat.
Thank you.
Okay,
Bye.
ciao.
Bye.
Thank you.
Bye.